And someone might check the chat that just came in. All right, I'm going to get things going here. My name is Keith Abels, and I'm the Soil and Water Specialist for the Sonoma Resource Conservation District. And I thank you all for joining us. And we're going to have a great workshop and event today. It's really exciting to have so many people that are participating and have shown interest. We had 386 signups as of this morning. So that's really exciting and unprecedented interest. I'm going to say a couple words about Zoom and Zoom etiquette. All participants are muted and your cameras are turned off. We'd love to see your faces, but doing this helps to ensure that the symposium will run smoothly and with limited interruptions. To remind everyone of webinar etiquette on Zoom, you have two options at the bottom of your screen. They are the Q&A and the chat. And the Soil Help team will be monitoring both, but make sure all questions that you have for speakers and panelists go into the Q&A box. They won't be answered if they go to the comment box. Our Soil Hub team will do the best to answer questions on the fly if they can, and then catalog the rest, and we'll answer as many as we can after each speaker's talk. Any comments, thoughts, or anecdotes from the field that you would like to share with the group can be added to the chat box for all to see. And also keep in mind the bios for each speaker will be posted in the comment or in the chat area, so you can learn more about them there. A quick few words about the Resource Conservation District, which we call the RCD. This is a North Coast Soil Hub event, which is a collection of 4 RCDs, Gold Ridge, Napa, Sonoma, and Mendocino. And you'll hear more about the hub at the latter part of this event. And we'll go into detail about our projects and what we're doing. But the RCDs help with soil and water management and conservation. We bridge landowner and production needs with resource concerns and environmental concerns. So we help people with designing, planning, implementing. We're responding to the fires and doing that type of work. Soil health has been a big focus for several years for us now. And this is part of that endeavor. We help in a number of ways. I encourage you to look at the website of your local RCD, or if you're logging in from another area and they're not called RCDs outside of California, you might want to look to see what the equivalent is there and get in touch with them. They can help you with soil and water management and conservation. And just keep in mind, we're here for technical assistance. We're not a regulator. We're here to help you as we can to manage your land the best as possible. I'd like to thank the Natural Resources Conservation Service, NRCS, for helping support our Soil Hub and this event. And I'd really like to thank the presenters and panelists for generously offering their time and expertise. Today, the title is Climate Change and Soil Health, Global and Regional Perspectives. It's really exciting today because this is our 4th annual soil health event with a vineyard focus. And we've been doing them in Sonoma County with some other, regional participation, but it's really been more of a Sonoma event. And we've had great turnout, but we're able to expand. And though it's unfortunate we can't meet in person, we're getting a lot more people that can participate in the Zoom environment. And that's really exciting. And it's really exciting that we can get some really good folks and expertise from other regions and other areas, because we do a lot of events and we tend to hear from people in our region, and it's nice to reach out to new people and hear new perspectives. It's kind of unbelievable how much soil health interest there is. I've been organizing a lot of events along these lines of environment and resource concerns and agriculture since before 2000, maybe helped organize up to 100 events. And when we put on these soil health events, we get signups quick and easy. We don't have to do crazy outreach like or intense outreach maybe for other subjects that sometimes we do. There's a lot of interest in this, and that's really great. And I think part of that is because soil health is so key to production. And it's so key to growers' bottom line. If you have good soil health, you have healthy vines, you have strong production, you can make more money, and that is the name of the game for everyone. Soil health also, the same practices that create soil health are beneficial to the environment. They sequester carbon, they reduce greenhouse gas emissions. And we're talking about the basics, which is really nice too. Soil health, really, when we're talking about it, we're focused on cover crops and how you manage them, compost applications, reduced tillage, and to a lesser degree, woody plantings like hedgerows and riparian buffer enhancements. But these first three I mentioned are all the basics of anyone's farming operation, and everyone has to manage those things in some way. What's unique and I think really special and a real opportunity with this is that the practices that benefit the environment and are also benefiting your agriculture practices and production and your bottom line are really, they're practices that are basic practices that are not necessarily something that are an additional cost and burden. They directly benefit you. Yes, it takes resources and to apply compost, for instance, but it directly benefits you, and it's not an added project like that could cost a lot of money and require a lot of resources that don't necessarily help your bottom line. So that's really unique, because a lot of the environmental practices are additional activities or require additional resources. And like I mentioned, soil health is key to production, and it's key to sequestering carbon and reducing greenhouse gas emissions. I think one thing we can all agree on, if you're in the agriculture business or really just observing in general, is that we're seeing changes in the climate, we're seeing changes in the weather, and we're seeing more extremes. Certainly here, we're having a pretty intense, we see another drought year coming into play with the limited rain we've had this year. But in general, we're just seeing more intense weather events and changes. And so the question is, how do we adapt to that? How do we respond to that? And so today is really about understanding what's happening, what are the trends, where are we going, what's been happening, and then what do we do to respond? How do we adapt and mitigate? So that is really the focus of today. I want to say today is more focused on the big picture, and we're looking more from above. We're hearing some more academic perspectives. And although we're also, some of that is it's definitely connected to the ground, but we're looking more from above, what's happening in the big picture. Tomorrow will be more focused at the ground level and what's happening in the field. And we'll hear from more growers tomorrow. So we're really lucky to hear from experts from other regions, as I mentioned, and help us to understand what is going on and what are some of the ways we can do, what are some of the things we can do to respond. So I just want to remind folks, and some of you are still logging on, maybe didn't hear my Zoom talk at the beginning, but if you want to ask questions, please make sure to put them in the question and answer section, which is in the bottom tab or the bottom menu on the right. And if you want to make a comment, but there's a lot of people that are participating today. So hopefully we can do as many as possible, but I can't guarantee we'll get to them all. But do put your questions and answers there, or your questions. So we're very lucky to have Dr. Greg Jones as our first speaker. He is very knowledgeable on climate and viticulture, and he is the director of the Evanstead Center for Wine Education and a professor and research climatologist at Linfield University in Oregon. So thank you very much, Greg, for participating. And with that, you have the floor, and let's hear from Greg.
Okay, thank you very much, Keith. I really appreciate it. First of all, can everybody hear me okay?
Yes, you sound great.
Okay, thank you. I'm going to go to sharing my screen, and I just need to confirm that as well. Can everybody see the screen with an opening slide?
Yes, you're all good.
Thank you very much. So Keith, I really appreciate it. Everybody else that has been involved with putting this event together. It's a pleasure to speak with you today. As Keith said, I'm going to be talking about a very broad kind of general look at this issue from research that I've been doing since the late 1980s, early 1990s, and when I did my PhD in Bordeaux. I've been collecting data and looking at climate, viticulture, and wine for these many years, and I'm going to share with you some very general things that we've been finding and understanding out there. So the first piece, just my outline real quick. We're going to talk about very general climate, viticulture, and wine production connections, talk about some observed changes in climate that we're seeing out there, what future climate change potential has on this industry, and then summarize all that and talk a little bit about some adaptive potential. And much of this, I think, is really going to come out in the other sessions that you're going to have later today and tomorrow as well. So from a big picture perspective, I think everybody on this webinar here understands this, but I'm going to go through this a little here. First of all, weather and climate really presents some very distinct spatial and temporal scales of influences to viticulture, and also, of course, risks. Individual weather events that tend to be short-term and localized are very important. Climate variability that's seasonal, decadal, and regionalized is very important. And then climate change and structure, long-term, regional to global kind of frameworks behind how the climate is suitable to individual crops, especially viticulture, is very important. So when we look at this as a perspective from a climate scientist and even from growers within the business, we see these as crop risk, production and quality variability factors, and then also this broader long-term suitability. So I'm going to talk about a lot of these from a very meta-analysis kind of perspective, but I'm going to start off talking about the suitability framework, something I've done quite a bit of. I drew this diagram on the back of a cocktail napkin in a bar in an airport back in early 1990, and it was the basis of kind of my PhD, looking at climate metrics and different wine production and quality metrics, trying to figure out kind of what do we know and what do we not know about this whole relationship. We know that the relationships are typically parabolic, that somewhere along the way there's too cold of a threshold where you just can't ripen the fruit. Your sugar levels are too low, you get unbalanced fruit. We also know somewhere along the way there's too warm of a threshold where you have lower retention of acids, you typically get overripe flavors, and again, you're unbalanced overall. So somewhere in this middle optimum zone is that Goldilocks, the thing that we all look for to have very consistent ripening. You get good flavors, you get typicity from a given variety. We know that there's vintage variations that drive some factors in there in terms of individual risk, but this idea of this optimum zone is what I've been really trying to look at. So this idea that we have these thresholds, can we understand them? And in doing so, one of the things I think is really important, how much plasticity or adaptive potential do we have within the system in the face of climate change from a management perspective that may be short-term, or a varietal perspective that could be much more long-term. So to give you some factors associated with this diagram is something I've collected data from just hundreds and hundreds of places that grow these varieties, trying to put these thresholds on them in terms of just their growing season average temperatures. For those that use growing degree days, this growing season average temperature is correlated at 0.99 or more worldwide to GDD. It's easier to calculate and it's a simple temperature value. And you can see the bands here in terms of these optimum kind of thresholds that we find out there worldwide. For all varieties, we know that there are some range of temperatures that they can grow in. So there's stylistic differences that can be found across those ranges from whether a variety is grown in a cool versus a warm climate. In a cool climate, a variety like Tempranillo could produce lighter body, lower alcohol, higher acidity, and brighter fruit. Warmer zones can produce fuller body, higher alcohol, softer acidity, and darker fruit. So I think everybody understands this. This makes a whole lot of sense. We know that varieties can be grown outside these bounds, but they're typically not going to produce economically and or quality that will be worthwhile to the grower. We can talk about Pinot Noir. It's just a good example. It's also the #1 variety up here where I live, but it has one of the more narrow climate niches in this grouping of varieties for quality production. From what we know about today's Pinot Noir regions, growing season average temperatures tend to be 57 to about 61 degrees. Therefore, it has about a 4-degree Fahrenheit climate niche. We know that it can grow outside those bounds, but not in a very high quality and typicity kind of framework. Burgundy is a great example. The vertical line here is the 50-year average climate for Burgundy, and the horizontal bar with arrows there is the 1.5 standard deviation of that climate over the past 50 years. And you can see during the coolest of years, Pinot Noir can ripen, but it's a lighter style. In the warmer years, it tends to get a little bit bolder and a little bit different in style. So that's just one perspective. And we can also put some quasi bookends on this here. The Tamar Valley of Tasmania is one of the coolest Pinot Noir producing areas today. And Russian River Valley and maybe a few other places are one of the warmer areas for Pinot Noir out there today. You can also take this kind of information and look at where these places are. And this is some of the work that I've been doing with this. So just looking at cool climate viticulture for cool climate varieties, averaging 55 to 59 degrees Fahrenheit, we can take a look at that and then talk about how climate changes will shift some of this information. So when you look at this, this is a map. This data is a little bit old. I need to update it, but this is a 50-year prospectus on growing season average temperatures, and it's a split map. So it's a growing season in April through October in the Northern Hemisphere and October through April in the Southern Hemisphere. And I know it's kind of hard to make out some of these things in here, but let's go ahead and drill into, let's say, Europe. Everything in purple are places that in theory would be suitable for cool climate viticulture. And you can see some of the natural places that we know in parts of Germany and Hungary and Austria and northern France and even Galicia. But you also see some places that potentially don't grow grapes. And the whole idea there is that there are other weather risk factors that may limit them. You can also drill into the United States, and again, a similar kind of map. And again, I'm not saying here that grapes, Pinot Noir or any of those kind of cool climate varieties are being produced across the Dakotas. It's just that that's where the suitable area is if there wasn't other risk factors. And you can see some of the characteristics of this, especially the valleys up in the Okanagan, and the ring of the valleys around the Columbia, the Willamette, coastal zones in California, so on and so forth. So now what I want to do is I want to move from this kind of broad perspective, looking at kind of general suitability, and talk about a meta-analysis looking at a cycle of vine growth, going from harvest in one year over winter through bud break, flowering, veraison, and harvest to the next year. These are the types of changes that have been seen worldwide. Number one, warmer dormant periods have been seen virtually everywhere. This has tended to reduce chilling and cold hardiness. And even though grapevines don't need the same amount of chilling that some horticultural crops do, it still is a requirement to have very even bud break and flowering later on in the year. But the issue here is that winter freezes do not go away in a warmer world. They still occur, they just occur with a different frequency. And I think in some ways, the plant system and the human system are less prepared for them. We also get to the spring period. We clearly have seen warmer air temperatures, but we've also seen warmer soil temperatures. And given the fact that I'm talking to a group of people that do a lot of soil work, I'm going to challenge everybody out there to get temperature probes in the soil everywhere you have a weather station. The reason is that you can model bud break and model certain aspects of spring growth if you have soil temperature in addition to air temperature. Very much important, but again, we've seen places that we do have data. We've seen warmer soil temperatures, warmer air temperatures, and earlier growth in the year. But again, frosts still occur. So we still have a risk there. It's just, it occurs with a different frequency. As we move a little further into spring, we've had some evidence of shifts in rainfall and cloud cover that has impacted flowering and berry set in some regions, but it's a little bit inconsistent overall. It's just that there are some evidence of that. If we move further into the summer, We know that the core area of the growing season has actually gotten longer pretty much everywhere worldwide. We know that there's higher heat accumulation, but we also know that there's higher temperature variability. The within season standard deviation of temperatures from week to week has actually gone up in many places worldwide. And there's also been more heat stress, a change in heat stress events in terms of both of their severity and duration in many regions. When we move over toward harvest, I don't have a lot of time to talk about this, but what we're seeing is lower diurnal temperature ranges because minimum temperatures are going up a little bit faster than maximum temperatures in many areas. So the diurnal temperature range, which really helps for preserving flavors and aromas in many varieties as they ripen, is just changed quite a bit. We also know that typically warmer regions are causing fruit to ripen too early. We tend to be very sugar ripe, we lose a lot of acid, and we tend to not have flavor and aroma in sync. So this idea of ripeness clocks being out of sync is something that we have to manage as we're growing grapes for wine production today. Again, I don't have a lot of time to go through this, but just to give you a kind of a meta look at it, all of the data that I've worked with, and I've worked with hundreds of varieties in dozens of regions. We've seen on average about a 4 to 8 day earlier occurrence per degree Fahrenheit of warming. We've also seen that the phases have become compressed, where some are changing more rapidly than others. And so the framework behind that is it just is more rapid within the plant system within a general growth cycle. This is the thing, I can't talk about all of these, but I know from the agenda for the rest of this meeting that these things will be discussed. There are clear changes in soil fertility, salinity, and erosion happening in different ways in different places, whether it be in Australia or Chile or Argentina or parts of Southern Europe. All places are seeing some kind of impact here. We also have CO2 fertilization, but while we understand kind of what that really means for the plant system, we really haven't done much research in the wine sector to understand what does it mean in a higher CO2 environment for wine. Of course, a lot of what you're going to talk about in this meeting is water availability and timing for irrigation if it's possible. The shifts in this are really important for this industry and all of agribusinesses. And then a whole slew of different issues associated with how disease and or pest timing and severity have changed in different places. Some of it because of climate, but some of it also because we just move things around too freely sometimes. So just to give you a sense of some trends that are out there so that you can get an idea, we did this project a few years ago. It only goes through 2017, but we looked at these 23 locations around the globe and we're trying to assess kind of how temperature trends were occurring in these locations. And so these were cool climate regions all the way to warm and hot climate regions overall. And if you look at some of the trends, I just threw a few examples in here for Surrey in England, Burgundy, Napa, and Perth. Very different kind of climates overall, but you can see that the trends are just over 2 to just over 3 degrees Fahrenheit. All of these locations, all 23 of them, warm significantly with decadal rates of about 0.22 degrees Fahrenheit. The period of record averages across all 23 regions is 2.5 degrees Fahrenheit. So again, remember what a climate niche for a variety like Pinot Noir is. It's only about 4 degrees. So if you're warming by 2.5, it depends on where you are in that 4 degree window. I thought I'd put something here in California. This is California's June through August averaged across all divisions. The trend is roughly about what we've seen in many of the places globally, about 2.5 degrees Fahrenheit. And all climate divisions in the map on the right-hand side have warmed during the growing season from about 2 to 3.3 degrees Fahrenheit since 1895. We can also go to precipitation. This is something I'm not going to get in greatly with here, but I think you're going to talk about it more in the conference. But look at these regions that we're looking at, ranging from many places all over the globe. If you look at annual precipitation, it ranges from 8 inches to 48 inches. So what does that tell you about how much rainfall is needed to grow grapes and make good quality wine? It's quite variable. The other interesting piece of this is growing season precipitation also ranges quite a bit, from about 3 inches to 22 inches in general. And if you take the ratio of those two, we find growing season precipitation to annual ranges from about 10% to over 84% in Mendoza, Argentina, where they get most of their rainfall in the summer during thunderstorms. But again, somewhere at about that 45 or so percent rainfall amount is during the growing season is where irrigation becomes much more almost necessary. So in terms of trends, there's very few precipitation trends across this whole set of data here. Lots of interannual variability in most of these regions. And so not quite the same kind of signal that we see in temperature. California, again, 1895 to 2020, and this is annual precipitation. There's really no trend. There is some trends in some seasons and some divisions, but it's not a real clear signal. So I think the bigger thing here is that what kind of drought structures are we either in or not in overall. So climate change for the future, again, I'm not going to get into a lot of great detail into this, but I'm going to go to this diagram right here from Berkeley Earth. And I think what's important here is that if you go from about 1970, 1980, the change in global temperatures has been almost linear. And some people even think that it might even start becoming a little bit more exponential. But the basic framework is if we take that signal from 1980 and go out to mid-century, we're really looking at 2.7 to 3.6 degrees Fahrenheit of warming out to mid-century. And this is a global kind of perspective, but you can see that there would be some variation from place to place. We can go back and look at this diagram again, a split map looking at places that are now too warm, that are in purple, warmer but still maybe suitable for cool climate, or completely new areas. And again, I know it's hard to pick this out, but let's go to Europe real quick. And I hope the colors come through for everybody here. But everything in purple now becomes too warm for cool climate viticulture. And then again, moving into the pink areas, this is why the English industry has gotten better over time, because we've seen this ability to ripen fruit when it couldn't do that not long ago. We can go to the United States, and again, a very latitudinal kind of shift in terms of new areas, a little bit of shift toward the coast, a little bit of shift up in elevation as well. So you can see these changes that are potentially out there. I can also show you some research that we did, and I think this is a really important aspect of this. These regions in Europe, these are 18 regions that we did where we used the random forest model to look at or train up the models to know what the climate, landscape, and soil characteristics were in these regions. And then we said, okay, let's fast forward the climate and see how they change. So everything that you see here, the darker areas are the boundaries, the approved AOC, DOC boundaries in these regions. And all of the little speckled darker spots are actually land cover for vineyards. So if we take this and we train it up for a warming to mid-century, what ends up happening here is that some of these regions find that their boundaries are now probably outside of what traditionally they have been known. I mean, there's a lot of really good examples. If you go down into Portugal, here in the Tejo, Ribatejo, most of that area moves closer toward the coast. Parts of the Douro get scattered all over the place. Areas of La Mancha scatter a little bit. So you can see that the idea is that there are shifts that happen both higher in elevation and typically more northerly in latitudes as well.
Greg,
Yeah.
you got 10 minutes, okay?
Yeah, I'm doing good. I've just about got it all down pat here.
Great.
So I'm going to shift and just look at some distribution data, and this is for the Willamette Valley, and this is our growing season temperatures. And I apologize, I have degrees Celsius here, but 1.1 degrees Celsius is about 1.85 or so degrees Fahrenheit. And so this is our 1895 to 2018 distribution overall. But I wanted to show you what happened for our climate normal from 1981 to 2010. We were shifted roughly 1 standard deviation to the warmer end. Basically, that whole distribution shifted pretty consistently. If we look at 2020 to 2040 in terms of conservative modeling, we're projected to shift again almost another standard deviation. And if we look at 2040 to 2060, moving up to being close to 3 standard deviations above where we have been historically. The interesting piece of this, and maybe this will come out from others speaking in the next couple of days, but one of the things I find very interesting is 18 of the last 20 years have already been here. So the question is that has our climate ramped up even faster than our modeling efforts are projecting, or are we just seeing a really a sharp warming now, and maybe it'll come down a little bit and then go back up? It's really a hard call, but I'm concerned that we've already been where we think we might be by mid-century, and that our modeling may need to be relooked at to figure this out. So going back to this suitability, what does a changing climate mean for a given wine region in terms of just what they can do? I'm going to go back to the Willamette Valley. We've already seen significant warming from, if you think about the 1961 to 1990 period, kind of a baseline, this was our climate envelope in the Willamette Valley, meaning from the warmest to the coolest places. That's what our envelope has been. During 81 to 20, this is how it transitioned. During 1991 to 2020, which is our new climate normal, this is what I've estimated it's going to be coming up. But then if you go to mid-century, that's what it's projected to be. So what that means is that if you look at the coolest of places within our region will be those places that are remaining in the understood climate suitability window for Pinot Noir. So it could mean pretty dramatic kind of shifts overall for us thinking about how we're going to be suitable for something different. So from a summary perspective, and tying this a little bit back, I've mentioned much of this. The planet is clearly warmer than any time in the past that we've had recorded data, and continued warming is highly likely. Persistence in the climate system tells us that. It just, we don't have much that's going to change things. I haven't talked about this a lot, but I think this is huge. The oceans are absorbing much of the heat, disrupting biogeochemistry cycling and a lot of the feedbacks that we have with climate. As evidence, the North Pacific has been much warmer than normal over the past 5 to 10 years. Climate variability has also increased. I didn't show a lot of this, but we've had much more record extremes, but on both ends really, but much more on the warmer extreme side. And there's been increased severity and frequency in some aspects of the climate. Models project continuing increases in variability on top of warming. So we really have an added risk factor on top of average changes in climate. So just some summary here. If I did a little meta-analysis, we've seen roughly about a 1.5 to 3.8 degrees Fahrenheit of warming in the last 50 to 100 years in wine regions. Precipitation trends are highly variable, as I mentioned earlier. Grapevines have responded. They're showing us that the climate is warmer. Phenological characteristics typically have accelerated and changed. We've also looked at some of the modeling efforts that are going on worldwide. And there's roughly somewhere between 2 and 6 degrees warming projected in wine regions. Conservative estimates tend to put that at about 2.5 to 4.5 degrees Fahrenheit, being highly likely. And again, going back to this issue, however, during the last 15 to 20 years, we've already seen conditions that were expected to be more like on average by 2020. So what does that really mean? Some challenges are out there, and I think this will be discussed overall in this conference in the next day and a half here. Changes in vine growth and productivity are clearly out there. We've seen this. Changes in heat and drought stress. Matter of fact, modeling in California says that heat events are 2.5 times more likely in the future than they are today. And the swings between really dry versus really wet periods are likely to be more frequent as well. Changes in pest and disease occurrence and severity is clearly happening. Changes in ripening characteristics. have been observed and documented pretty much everywhere, which forces us to really think about what wine styles can we produce. Water resource issues, big one, and I know this will come out over the next day and a half here. Water resource issues are going to be a real challenge in the West, especially. Most large wine regions in the world today are in dry summer climates that depend upon winter snowpacks, and we're going to be challenged, I think, in that arena. Long-term varietal suitability is clearly an issue, and I think we're going to see more of that. And I think one of our biggest issues is trying to understand how to increase adaptive capacity while reducing vulnerability. And I'm going to give you a summary of the things that I have seen in research in other regions in terms of what this industry is trying to do. Number one, we need to utilize the wealth of cultivar diversity in Vitus Vinifera. You know, we grow roughly 20 of some 5,000 different varieties. And I think we're missing the boat in really understanding what might be very suitable and good fruit and therefore wines. The problem is there's a marketing issue. I know you're going to talk about that as well. As such, we also need to realize the genetic potential for adaptation as well. We have large landscape potential also. And I think people are starting to realize this more and more as we plant new areas or replant old areas. Again, a big conversation, I know Keith mentioned this, and I know you're going to talk about it, is developing regenerative agricultural approaches, being much better stewards of our landscapes, our soils, and our biodiversity within and around vineyards is going to be very, very important. Optimization of training systems, whether it be the canopy geometry, orienting rows in new vineyards, or even using shading materials. A lot of this kind of work is going on in many regions, and we need to do more of it. This is something I think is worth mentioning here, especially as we talk over the next day and a half about soils. I think we need to really better understand the right combination between the scion and rootstock that we're planting in a given region, soil, and climate. I think we know quite a bit, but boy, I think there's a lot to learn here that would be very beneficial for us in the future. And then lastly, understanding water use efficiency and how to manage irrigation where we have it or where it's possible. I think these are all kind of very big things that we need to definitely learn more about and be able to do to produce a more sustainable wine sector. So with that, I think I'm kind of almost right on time. I did want to mention one more thing here. Up in the upper right-hand side, there's a link there for monthly weather and climate reports. I've been writing these for about 20 years, and it's just something I do. It keeps me totally on top of what's happening. It's very much a West Coast perspective, California, Oregon, Washington, Idaho, trying to document what's happened both in the current month or current season, but also look at short-term forecasts. So if you're interested in that, you can go to that link and sign up, and you'll get put on my my email blast for that. So with that, I want to thank you very much, and happy to take questions either now or later, however you guys want to manage it.
Great. Thank you so much, Dr. Jones. That was an excellent presentation and very succinct. And there is a lot there to think about and absorb, but a lot of practical information. So I want to remind folks to put your question and answer. We have a nice chunk of time here. We got 15 minutes to do question and answer. Put that in the, if you look at the bottom of your screen on the far right bottom, there's a Q&A tab. Please start entering your questions there, and do not put them in the chat. They won't get answered if you put them there. But yes, thank you very much. That really set the stage for what we're talking about today, and from a viticultural perspective. With that, we're going to have Katie Brantley, who is with the Mendocino RCD, lead the question and answer session. And take it away, Katie. Thank you.
Great, thanks, Keith. And we have quite a few questions already rolling in. I'll start out with one that's asking, are growers already planting different types of grapes that are more suitable to warmer climates in preparation for this continued warming trend and looking ahead to the future?
Well, I'm going to go back to Bordeaux real quick. I think everybody, if you've seen the news in the past couple of years, Bordeaux has been working toward approving new varieties for that region. And you know, in Europe, that everything is pretty well controlled in terms of what can be grown where and how. But I go back to a talk I gave in 1994 in Bordeaux. I told them that they needed to be doing this, or at least thinking about it. And it took 25 years to do it. It's not that I want to be right or wrong in this whole game. It's just that, you know, it takes time for people to, especially in places where there's a very identity-driven kind of issue with varieties. But I think in the rest of the world, New World, we actually have seen a lot of people experiment more. And I think it's really important that why plant all one variety in your vineyard when you can plant a small test plot so you know what other varieties can perform. I think it's a natural thing to do. So people are doing it. It's happening slowly. Here in the Willamette, we have definitely seen more varieties being tried up here that historically could have never grown here 25, 50 years ago.
Great, thanks. Okay, next one. So your Willamette climate overall varietal slide was very interesting to folks. They're wondering if you have something similar or have at least some information to share with us today about Sonoma and Napa County regions. I would say any of the North Coast, California.
I don't have it in this current slide deck, but you know, it's pretty easy thing to do. If you take that varietal framework and you calculate out the climate envelope for Sonoma and Napa, you can map that out. Of course, Napa is a little bit on the warmer end. It typically has a wider climate range, but covering a large collection of the warmer end of some of those varieties. I've given that talk somewhere before, and maybe when I put together a PDF of this to share, I'll put it at the end of that so you guys can see it.
That would be wonderful. Thank you.
Mm-hmm.
So we have another question that's, I'm curious what your response would be to this, but they're wondering if you have results that more or less cover horticultural crops that might also be in some of these regions too, and if you're seeing similar trends.
I've done some work with colleagues on a couple of very large projects in Europe, looking at olives and wheat and almonds in terms of broader kind of climate structures, suitability and change in Europe. I haven't done as much here in the United States, but I would imagine that similar kinds of things can be done for olives, for example, in California. I have been talking with individuals in a kind of a worldwide coffee federation that we're looking to try to define what are the climate boundaries for different types of coffee varieties as well. So I could imagine this could be done for other crops in California.
Great, thanks.
Mm-hmm.
This hopefully will be a quick question, but this is regarding your variety temperature range table. They're wondering if the average temperatures are 24-hour ranges or daytime, growing season, or annual.
Sure. Thanks for the question. So one of the reasons I used average growing season temperatures is that over the years, I have found how erroneous and problematic growing degree days are. The issues with growing degree days are that it's often confused when you say the term, people don't really understand it. They think it's a temperature. I know people in the industry do, but in general, it's a misunderstood factor. The other thing is that when you calculate growing degree days, if you don't do it correctly or your weather station software is doing something different than you think it is, you can't compare your data to somebody else's data. It's just not very feasible. In the American Journal of Enology and Viticulture, we published a paper in 2010 which defined the Western United States temperature characteristics and all of the problems with growing degree days. So all that being said is that when I started to do this, I tried to go to something that was simple, which was just temperature. So all I'm doing is taking the 214 days in the growing season. And you could actually do the growing season March to September or April through October, and you get roughly the same results. But it's essentially a 24-hour average temperature and then averaged over the 214 days in a growing season.
Thanks, that helps me understand it better too.
Mhm.
Would you expect oceanic and coastal warming to happen at about the same rate as warming inland?
Yeah, I don't. I think our data so far has showed us that on land warming has been a little bit higher than oceans, but it hasn't. It's not dramatically different. I think the challenge with if you look at the west coast of the United States, the warming that we've been seeing, it does a few different things. Number one, a warmer ocean produces more humidity, basically more evaporative potential coming out of the ocean. So that's why nighttime temperatures have a tendency to be just a little bit higher. But it also produces higher disease pressure when we probably don't need it overall. The other thing that happens, and I think this is really important for the area of the North Coast and the Bay Area, is the dynamics between coastal upwelling and colder water along the coast and the day-night cycle of fog. Our modeling is trying to answer some of this, but it's a real complex issue because the fog is the result of the inland areas in California warming. So warm days, you tend to have rising air that helps pull in the fog. But then overnight, the fog backs itself out. And, so the issue comes down to is that what does the interface between the either warmer Pacific, maybe a narrower range of cold water near the coast, and a really warm inland do? Does it make the fog more prevalent or less prevalent, or occur over a narrower area along the coast? Those are all big questions that haven't been answered yet.
Obviously, lots more to study.
Mm-hmm.
Are you seeing trends in overall vineyard lifespan and replanting that's kind of related or parallel to these climatic trends?
Well, that's kind of a hard one to call. I do know that a lot of people are looking at clearly much more holistic regenerative processes within the vineyards, and I think that in and of itself would be what would create a greater lifespan within vineyards. We have a lot of different challenges, whether it be heat and drought stress to pest and disease severity that potentially are putting more pressure on some of the health of vineyards today. But I think an overall holistic approach would be probably the best thing for us to do in the future.
We should get to hear more about that with some of our other speakers too.
Sure. Yeah.
How do these climatic changes impact the soil organic matter in vineyard soils?
Well, you're just talking about more rapid recycling of material near the surface. And again, I think that's why regenerative farming is such a better way to go. So we can maintain some of that a little bit better. The wholesale kind of removal of surface vegetation is likely in many ways not the best way to go. And so if we can kind of maintain a more biodiverse kind of set of plants and animals within that continuum, I think we're going to be better off in general.
Great. So you mentioned the importance of monitoring soil temperature as it relates to bud break. Is there a model available to interpret the data collected?
Actually, and again, great question. And I'm going to challenge everybody to get more data on this. There has not been a kind of universal model developed in this framework. The data that I have looked at, and I know other people in parts of Europe and Australia have looked at, there's a connection between the soil temperature and the air temperature that can help understand when bud break occurs. So I just go back to a very simple plant system. Think about daffodils. Daffodils respond to a warming of the soil, but also a little bit of a day length kind of component. And so tying that warming of the soil back to changes in day length and changes in atmospheric temperatures is where I think the soil temperature modeling needs to go for bud break and even for bloom. Because as the soil warms from, let's say, mid-March right about now into, mid-May, I think if we have enough data, we can build those models. But the limited data that I've looked at, it's very promising.
I just follow up on that. Are there ways to share data that in a more timely fashion that might help with building those data sets?
Well, yeah, that's a good question. I think the scientific community has done a decent job at this, but has probably a lot to learn and grow from on it. I think we need to do a better job of collecting and sharing data across companies and businesses and governmental agencies. I've been working, I know some of you are probably familiar with Fruition Sciences. I've been working with them for the past 5 or 6 years, and I think that there's some really good work going on there to help try to better understand aspects of this and how the plant's responding to both climate and drought stress and other factors. And I think there's a lot of sharing that's happening within that arena. I don't personally have a place I can tell you just to go to share the data, because it really, I think there's some other ways in which we need to develop that system before we can actually make it be universal.
Sure. Well, hopefully we can keep working towards that.
I agree.
Are you seeing any climate change impacts on wine quality?
Well, it depends on what you determine wine quality to be. That's a big open question. How do you measure wine quality? Number one, there really isn't a universal number. I mean, we can take some kind of chemistry from the fruit, from the wine, and put it into perspective. You can also take certain things like, and I know this is opening a can of worms, but vintage ratings, how people have rated wines over time. And I've done a lot of work on this because it's really one of the only measures we have to try to look at this characteristic. And we've developed some modeling that has taken rating systems, not just one like Wine Spectator, but dozens, and puts them all together into what's called a consensus ranking. So we get the consensus of a whole bunch of individuals in terms of quality. And from that, we can really kind of look at some of the factors driving quality. And we've clearly seen some changes, but we've also seen some variability there as well. We've published papers on Burgundy, port wine, Tuscany, and Bordeaux. And at some point in time, I'm going to do something on the North Coast with the same kind of data. But again, it gets back to this issue. We all need to come to and understand what is wine quality, and do we have a better measure of it that we can use? Because that's the kind of things that we need to do from a climate science perspective to be able to analyze. We need really good data defining wine quality.
Makes sense. So getting back to the importance of measuring soil temperature, where and how deep would you recommend doing that?
Well, this varies a little bit. I've seen quite a few different numbers, but most of the time, and I'm going to talk in centimeters because that's what most people have been doing, is 10, 30, and then 1 meter or 100 centimeters. You don't have to do all three, but you should at least do the 30 centimeter value, because I think that's where we've seen the most correlation with plant growth.
I'm going to jump in and let you know we got time for 1 or 2 more questions. We wrap up this session at 9:25.
Great. How about, do you think looking at increases or decreases in microbial activity or respiration would be beneficial to track in relation to the grape growing and wine quality?
I really do. I mean, I think that's a great question. You really need to be able to track everything somewhat simultaneously. You need to track all that microbial activity, but also track that, in terms of fruit characteristics, and then tying it into how the wine turns out. Many research studies that are done often look at one thing in a compartment and then don't tie it into the other components. And so I think that as science develops projects, we need to be more critical about those projects being holistic, from, let's say, the microbial component all the way to the wine component. It makes projects more difficult, but those are the ones I think where we get the biggest information from.
Great, thanks. I think that's all the time we have. So sorry we couldn't get to everybody's questions, but please reach out to any of us, the RCDs, or to Dr. Jones directly to answer some of these additional questions that you might have. Thank you to everyone.
Yes, thank you, everyone. And thank you so much, Dr. Jones. That really was an excellent presentation and provided a lot of pertinent information. We are really fortunate to have another speaker with quite a bit of knowledge on this topic, and also very fortunate that he is farming down in Southern California in the Temecula area. So I think that's a good harbinger of where things are headed up here. And he also looks at things both from a viticulture perspective, but from a larger soil science and a higher view of things. So I want to welcome Greg Pennyroyal, who is with Wilson Creek Winery. He's the vineyard manager there, but he's also a professor of viticulture at Mount San Jacinto College in Southern California. Thank you, Greg.
Hi, Keith. Hello, everyone. Thank you very much for the invitation. Dr. Jones, that was a great presentation. We will be talking. I totally agree on the soil temperature thing. There's a lot of things that are changing rapidly. What I would like to focus on is, this is actually a really nice follow-up to Dr. Jones's presentation, is how do we get some of these concepts and how do you start to actually apply it to a farm? So I do have somewhat of a research background, but despite the owning of one clean shirt, I am a farmer primarily. We're doing about 170 acres here in the South Coast, in a region that has experienced a lot of heat in the recent decade. And so this might be a potential for what's going to be happening in the North Coast. So let me open up this presentation and go to the beginning. And there we go. So can everyone see the presentation properly?
We're actually looking at your presenter view instead of the full slide.
Let me go to, sorry about that. Oh, here it is. Use presenter view. Get rid of that. Try that one. Does it look better?
Perfect. Yes, looks great.
Right. So what I really want to talk about is with climate change, I think what we really need to look at is resilience. And you get resilience through biology. And I am defining the increase of biology as basically using the principles of regenerative ag. I want to talk a little bit about carbon sequestration in regenerative ag, and specifically some examples, some of the benefits I've seen as a grower, and some thoughts about how to get into this, because making a transition from industrial ag into regenerative ag can be a challenge, and you need to do it carefully. So, you're benefiting and you don't go bankrupt in the process. This is a field of echinacea up in Trout Lake, Washington. That's Mount Adams in the background. That was my first major venture into agriculture. Trout Lake Farm was the largest organic herb farm in the US at 2,400 acres. I later was the director of botanical research for Liner Health, which was a division of Pfizer in their natural products division. I spent 15 years with the US Pharmacopoeia developing standards for medicinal plants, which is part of the reason why winemaking was so interesting to me, is whenever we would talk about chemotypical expression of the microbiome in plants, I would inevitably get much better questions from winemakers than I generally got from pharmacognosists. Some of the research we've done is in the National Institutes of Health. I did some interesting work with the Global Institute of Tibetan Medicine on the same thing, looking at standardization of medicinal plants. I'm currently the vineyard manager at Wilson Creek Winery and a professor of viticulture at MSJC. I think Dr. Jones covered this really well. This is the South Coast mean temperature for the last 100 plus years. But if you look at this trend, it's it's clearly in an upward direction. And if you were to graph this, the rate is climbing quite a bit. The South Coast AVA is sort of similar to the North Coast AVA in that we have a lot of ocean influence here. Our diurnal shift is still pretty good. I think it's our saving grace, but as the oceans warm up, I think we might get a change in weather patterns. It's hard to predict whether it's actually going to get specifically hotter, specifically drier, specifically wetter. I think what's going to happen is the trends are just going to become more severe. The heat events are going to be hotter. The rain events may not be as much of a volume of rain, but we've noticed that the events are short and tend to be really hard rain. So let's do a quick comparison of industrial agriculture versus regenerative ag. So the basic theory is soil as a medium for delivering soluble nutrients to plants. That has led to a degradation of the soil food web. It also generally releases CO2 and decreases stable soil carbon. It does this through a degeneration of organic matter. This is a larger public policy issue, and I was glad that people, one of the questions that came up is, how does this work with other crops? I think as viticulturalists and anyone in agriculture today, if you look at the degradation of overall public health, a lot of that has to do with the drop of nutrient density in food. And that has a direct correlation with the microbiome and the biological expression of the soil that's feeding the plants. There's also an ecosystem services degradation. We're currently working with our local water district, is they had millions of dollars put aside to develop land for recharge. And my question to them is, why not just help the local vineyards create strong biological soils so the vineyards can be the recharge? A biologically active soil will actually clean water, and the water will enter the watershed cleaner than it came in. Also, industrial agriculture has an increased reliance on synthesized inputs, and that's pulling money out of your pocket as a farmer. You compare that to regenerative agriculture, is the soil systems are constantly improving soil health. It supports the food web. It sequesters carbon. It builds organic matter. It increases the possibility of nutrient-dense food as medicine. And that's sort of the connection that I have of having started in medicine and seeing the public health implications of loss of the ability of the microbiome to deliver nutrient-dense foods. And it decreases or eliminates the reliance on synthesized inputs, which will improve your bottom line. Which brings me to the issue of regenerative agriculture must also regenerate your farmer's bank account. If you don't have a good margin, you're not going to have a mission. It also must perform in current economic models. What I mean by this is there are a lot of models that are based on what's called true cost accounting, which basically says that if you're externalizing costs like water pollution, like CO2 loss from the soil and those things, that your actual economic models may not be as good as they look because those costs are put off into the general public. But that's not the system we're in. So it needs to work on those true cost accounting models. Hopefully when we get some correction of this with things like carbon credits, that this could be a help, and some of the benefits that a good regenerative farmer is providing to the community could be actually paid in cash. But until that happens, it still needs to work out. And that's the value of ecosystem services: clean air, water, open space, genetic diversity, and the potential for carbon sequestration. So if you want to change how you run your operation, I think the first thing you need to change is your perspective. So the regenerative model is that all plants have evolved in a highly complex mineral, organic, and biological mix. There's a theory of biological availability that basically says it's not so much that things are soluble, but they're made available biologically. There's some really interesting research currently going on under the title of rhizophagy, which is happening, I believe it is Princeton University. But anyway, it's an interesting concept to look up, and I would highly recommend it. Maintaining soil health is sustainable agriculture. You know, we hear these terminology issues of regenerative and sustainable. And I think the reason why a lot of us are going to regenerative versus sustainable is if you currently have a system that's degrading soil, that's losing water, that isn't increasing organic matter, that's not what you want to sustain. Most of us have inherited degraded systems to the point that we probably need to regenerate them, not just sustain a system that currently isn't working. The opposite of that is the theory of solubility. That's the basic chemical model. It's the NPK approach to fertility that assumes that the parts are greater than the whole. Large-scale commercial interests support this, and the challenge with this is it works. It's somewhat like anabolic steroids. They achieve great results, but only in the short run, and then degeneration and damage sets in after that. So this is sort of how I look at regenerative ag as the sort of core issue here. Is the photosynthetic is plants. We talk about soil health a lot, but the truth of the matter is that plants are driving the system, not the system driving plants. So plants functioning at a higher photosynthetic efficiency are the engine that develops soils, not the other way around. There's a lot of work that says, on average, most crops are working in their pure theoretical photosynthetic efficiency of about 15% or 20% of their theoretical genetic potential, which means that we're leaving 80% of that value behind. Now, of course, realize that unless you're in a greenhouse under perfect optimal conditions, you're never going to get to 100% because there's heat stress, there's drought stress, there's, you know, all sorts of biotic and abiotic stresses. But we can increase that ratio from 20% to quite a bit higher. And what happens when you do that? Well, it does correlate to increased yield, but it's not always just the yield. When you have increased photosynthetic efficiency, a lot of that efficiency goes into sugars that then feed the soil ecosystem. And this is a really important concept because the model that a lot of us learned years ago was that it's sort of a closed system, and the more inputs you put into the system, the more yield you get. Part of our yield has to be feeding the biological system that then sort of primes the pump. It gets the whole system going. The more the system is primed and going, the more it self-regulates. The additional sugars are improvement in the genetic expression of the actual vineyard. You get higher chemotypical expression, which is a technical way of saying you get more balanced fruit. Some of the examples of a more biologically balanced vineyard is the acid-sugar ratios tend to be a little more balanced. The nitrogen in the grapes also tend to be on the amine side. In other words, the ammonium and nitrates are forming into amines and then amino acids and more complex amino acids, which actually really help in fermentation. That's what creates a slow, steady, solid fermentation versus a sort of spike and crash that we sometimes see with other grapes. Improved plant health and resistant to biotic stress, excuse me. So part of that resilience is cover crops and mulches during the vineyard dormancy period. Vineyards are dormant classically when we get our Mediterranean winter rains. Don't let your crop, your soils be bare during that. That is an amazing time to collect solar energy and then feed it into carbon increase into your soil. Under the values of cover crops, this is a photograph of my Cabernet Sauvignon vineyard in last year. We had 15 inches of rain. You can see that the cover crop is a multi-species cover crop. It is pretty much up to the cordon. It has grown really well, and it is it's just producing a lot of organic matter and pumping a lot of sugars into the soil. This is that same vineyard this year where we've only had 2.2 inches of rain. Obviously, it's not doing as well, but with that little bit of rain I still have a cover crop. It's still covered. I'm still going to get mulch. I'm still going to get carbon out of it.
Greg, you're cutting out. You might
I'm
try.
not sure what happened with that.
Yeah, Greg, this is Keith. Your video is a little crackly. Maybe try turning off your video and try.
Okay. How's that?
We can see it.
How's that?
Yeah, you're still cut out. Maybe move to the next slide. Maybe that slide's an issue, or I don't know.
It says you cannot start your video because your host has stopped it.
Yes, I disabled your video, Greg. This is Ashley, just because we're having a hard time hearing you. So we cut out your video so that we have less bandwidth on your end. So go ahead and try and speak. Your screen is shared. We just don't have your
Okay.
face. So
Perfect.
for John.
Okay. Let me. Okay. So the bottom slide is a vineyard that we just started. We planted a cover crop. It has not been in a regenerative system. And you can see the difference. Even though the cover crop on the top one is obviously nothing like it was the year before, we're getting almost no growth out of the cover crop from something that wasn't in a regenerative system. So this is some work by John Kemp at Advancing Eco Agriculture. It's called the Plant Health Pyramid. And basically what it's saying is as plants become more healthy, they move up this photosynthetic pyramid. And what it says in the bottom is you get complete photosynthesis. On the next level, you get complete protein synthesis. As you move up this, you get more resilience. So at complete photosynthesis, you are building some carbon in the soil. At complete protein synthesis, you're getting a lot more expression of the plant's natural immune properties, and it becomes more insect resistant. The further up you move, the further the genetic potential of that plant is able to express in an ecosystem and able to express favorably with biotic and abiotic stresses, heat, insects, and all those things. Resilience to biotic stress goes up as you go up that plant health pyramid. I'm sure you're all familiar with the work of Elaine Ingham and the Soil Health Food Web, but this is basically what we need to consider when we're doing this, is as you increase the levels of plant driven complexity in the soil food web, the ecological homeostasis builds in. And what that means is the plant is communicating with the soil food web is feeding the plant, and you get that whole system going. For me, this is the goal of regenerative ag, is to get that system going. This is a classic plant succession diagram that says going from pioneer plants to a climax forest. I think as viticulturalists, a lot of us sort of forget, well, where does Vitus Vinifera fit in? And I think most people would say probably somewhere in the shrubs or woody pioneers. But the truth of the matter is that Vitus Vinifera is actually a climax forest ecosystem plant. It evolved in what's now modern day Iraq and Iran, and it co-evolved with oaks, using the oak as an infrastructure to get up to the sunlight, but also surviving in that fungally dominant, highly biologically active oak forest, savanna oak forest ecosystem. So with that in mind, when you're managing the ecosystem of your vineyards, you have to sort of ask, what are the goals? One of the tools that I use is to use a diversified cover crop to increase soil diversity. As my soils become more complex, I go from more simple mixes and more grass-based, oat-based mixes, more legume mixes, and I'm sort of pushing that, that complexity more towards the fungal diversity. This is a great tool that I use. It's called Smart Mix. It's from greencovercrops.com, I believe is the company. But if you look up Smart Mix, it's a great tool. You can take a look at what your goals are, whether you're trying to increase organic matter, fungal growth, nutrient cycling, you know, whatever those goals are, and you can play with different mixes, and it'll tell you how those mixes are going to influence your goals and what the cost per acre is, so you can help to calculate the return on investment for that. A quick note on agriculture and carbon sequestration. If you look at all the other forms of life, everything from amoeba to arachnids to humans to fish, this is the potential of carbon sequestration that they have as compared to plants. However, these aren't in the same scale. Plants have so much more of a potential to sequester carbon that if the plant scale was on the same as these others, it would look like this. That's how much plants have the potential to sequester carbon. But especially in warm soils and vineyard soils, you realize we're not just trying to sequester carbon. One of the goals of sequestering carbon is to lose it. You want that carbon to be recycling all the time.
Greg, this Keith, everything looks good. You got 10 minutes.
Thank you, sir. So I'm getting pretty much near the end. Keith, the taskmaster, is keeping me on target. This is mint field up in Oregon. Talk a little bit about what's worked for me. So soil tests, the right kind of soil test, especially with a saturated paste test, I think give you a good indication of where you're starting. I would not run my fertility program with a soil test. One thing I will add to this is we started doing the Haney CO2 respiration test. So far, the results of those tests much closer correlate to what I'm observing in my vineyard. Whereas the actual soil tests, I see a low correlation. Once again, that's more like potential. Ideal samples, I gotta say, I just really don't use them anymore. I don't find much of a correlation. Part of the problem with ideal samples is it's testing the entire tissue. It's not telling you what's in the sap, it's telling you what cumulatively has been in your tissue. As an early indication, early fruit set, possibly, but aside from that, I really don't use them anymore. A tool that I do use that I've been finding remarkable results is Sap Analysis. Some of the insights, so this is just a couple of slides of some of the Sap Analysis that we've done over the course of a year. And each one of those lines represents either an old leaf and a new leaf on a sample that's taken twice a year. A quick example of some of the things that we saw here is we saw the sugars early in the season being low and being quite erratic. And the question is, why am I not getting better sugar metabolism or photosynthetic efficiency? So taking an example of iron, we looked where we got a bump in the iron from one of the foliars, but then a drop, but then it increases as the temperature increases, and we started to do a different irrigation schedule. So the question was, is the iron not being taken up by the plant because of the soil temperature? And you would think that at higher temperatures, your soil temperatures are going up. But what happened at the point where these soil temperatures went up is I started irrigating on a regular basis in the evening. It was the slow feeding of the grapes, and the soil temperature probably dropped. So to connect with what Dr. Jones says is we're now putting out over 20 soil temperature sensors in our sensor base to take a look at that relationship to not only water uptake with soil temperature and not only phenological cycles of the plants, but the nutrient uptakes of the plants. Some of the other tools that we're using is a system called eVineyard. There's some tools out there, it's just the one that I use. The important point here is you need to have decent tools for data collection. This is one of the data collection tools that I really like. It's called EraWatch. It's a satellite-based technology. This is a 4th of July 2020. The red indicates areas where I have heat stress. It will give me an ETO reading. It'll give me a photosynthetic efficiency reading. It'll give me soil temp, air temp. It'll compare some of those. It has some algorithms that I can compare those. But what I like about this is a couple of these representations. So this is an overall stress. I can show this to my crew and we understand where we need to adjust the irrigation on that. As a comparison to this is that exact same field, December 31, after we had a rain, obviously really low stress, and it's picking up a lot of the photosynthetic benefits of a cover crop. The information that we got from the whole concept of soil temperature and possible oxidation reduction issues in nutrient uptake are leading us to have more control over our irrigation systems and possibly more plentiful fertigation in our systems. So this is a 4-acre block that we've divided up into 4 1-acre sections that we are going to be doing sort of a proof of concept that says if we are mulching this entire field and we are going to compare foliar feeding with fertigation and monitoring the soil temperature with the soil respiration and nutrient uptake. We want to know how those things correlate. Once again, this is more of a proof of concept because the controls I have are actually off-site controls, so there'll be decent controls for a proof of concept, but not exactly publishable material. So what benefits have I enjoyed? My increase in my 3rd year biological blocks have been 22 to 31% on average. But here's actually the most important part to me is yield stabilization. In 2020, the Cabernet Sauvignon in the Valley had a 65% of 3-year average yield. So in other words, the yield went down substantially. We do running 3-year averages. If you look back on 5-year averages, it's pretty stable. In the blocks that I had on a regenerative program, I still yielded 110% of my 3-year average, which is a little bit statistically off because, you know, obviously I'm getting an increase in yield. But the main thing to this was the yield stabilization. I'm also getting better quality. We're getting better ratings on our points. Our winemaker is happier with the fruit. And you can just tell, you know, as viticulturalists, when you're out in the field, you can just taste your fruit. You can see and taste when it's doing better. Part of the expression of that being better is this is a picture of Malbec, and you'll see that it has these large clusters and not any real small clusters. This is the expression of the primary buds and the apical buds. In other words, when the buds develop, they have 2 primary buds. Well, they have a primary bud with 2 clusters in each bud. When you see those large clusters, what you're actually seeing is the vine is expressing genetically that it has enough nutrients to express its largest clusters. I have a 50% reduction in the use of fungicides. My goal is 0. A 38% reduction in the gallons to ton. And I will actually say about 15% of that was due to infrastructure improvement. I can't separate those two, so it's hard to tell how much. I'm just guessing it's roughly half from regenerative and about half from infrastructure. Also, because I have this kind of consistency, I plan more, I react less, I have more field and crew time, which is why I became a viticulturalist. I have more customer time to spend with my customers. It's increasing profits. I'm having way more fun. This is so much more fun than the old system of like spraying on a schedule and, you know, being freaked out about trying to constantly correct problems. And I don't work on weekends much. You know, we still all do, but it's a lot less. So what's your tolerance for innovation? If you're an early adopter, you really want to ask why. A lot of people love innovation just for the sake of innovation. If you do it, be careful, do it on a small scale because it might work, it might not. I think most of us are right here. We're looking at that chasm between the early adopters. There's enough research to show that this is a good system, and you need to ask how. And the way you ask how is you ask for help. We look for systems, technology, and experience. And that includes folks like the RCD, the NRCS, Vineyard Team, Kind Harvest is another group. It's a web group, Regenerative Ag News. There's a lot of great resources out there. Whatever you do, don't listen to the coffee shop crowd, and be really careful about taking advice from your dad. How to get started. Have clear goals. For wine grapes, it's not always yield. One thing to keep in mind about yield and quality is it's a perennial crop. The first year of your program, you're probably going to see very little change. In the second year, you'll see significant change. In the third year, you tend to see stabilized change. It's like any improvement in a vineyard. It tends to be a 3-year cycle. For me, the big thing was less variability. And on the quality issue is I'm defining quality by how my winery defines quality. Better yields, of course, just make economic sense, but it's the quality of the grapes that is the ultimate determinant here. You also get a better lifestyle, better work balance, better community member, and better economics. I've been rich, I've been poor, rich is better. I suggest starting small, make mistakes on a small scale, look at your return on investment. Once you've got a small scale that works, scale up fast. Develop appropriate measurements. You don't have to measure a lot. Like I said, I'm a real fan of Sap Analysis, but if there are other things you can do in your farm, including just going out and being observant, that works. Make sure that you have a good record keeping system and you have good field observations. And in summary, I think regenerative ag may be the best system to develop resilience to climate change and biotic stress. It can perform both financially and environmentally. There is technology now that can assist. And there's also, I have to admit, I'm really impressed with what NRCS and RCD and the folks that are creating the infrastructure, how much they've embraced this and how ahead of the curve they are. And as a last thought, what we do to the environment, we do to ourselves, for better or for worse, and better is better. All right, so questions.
Great, thank you, Greg. That was a really good presentation, very to the point, and dovetailed really nicely with Dr. Jones' presentation. So thank you for that, and thanks for bearing with the technical difficulties, but in the end we could hear and see everything. So that worked really well. And my take-home message is that improving soil health it improves your grapes, your bottom line, and is fun. So I like that. With that, we have about 15 minutes for question and answer. And Katie, if you could lead that, take it away.
Sure, thanks so much, Greg. I think there's a bit of a clarifying question that might be helpful for everybody. Just a quick difference between industrial versus regenerative in some of these systems, I guess particularly the viticultural systems.
Well, you know, the industrial system is basically a system that happened post-World War II, where we started to look at soil as just a medium for carrying water-soluble nutrients versus the soil as a complex biotic system. And that's the primary shift. If you look at it as a biological system, your perspective just changes. You know, one of the reasons why salt-based synthetic fertilizers work so well is they pretty much kill all the biology in the soil, and that biology has nutrients tied up in it. And so you get this flush of soluble nutrients. Over time, the soil's ability to make those nutrients available drops less and less. And you just get into this negative spiral, as opposed to a regenerative or a biologically based system. You go into a positive spiral where the system not only is providing more nutrients, but it becomes more self-regulating. I'll give you two specific examples, vineyard examples. I had an old block of Chardonnay that just had a terrible nematode problem, a parasitic nematode problem. We just changed the soil organic matter, increased the overall biology with some compost and some cover crops. I introduced some beneficial nematodes and the problem just went away. I now have lots of nematodes, they're beneficial nematodes. The other one is I have an old block of Cabernet right against the restaurant that we really didn't want to pull out because it's a moneymaker as our Disneyland block, as we call it, the one that people go out and visit. And it had red blotch. And we just started foliar feeding and fertigating that and getting the nutrient levels up and moving up that plant health pyramid that I talked about before. And the red blotch stopped expressing. We just don't see the symptoms of red blotch anymore. I'm sure it's still in there. If I did a PCR, I'm sure I'd still pick up some red blotch, but it's not expressing. The fruit's good quality. It's fine.
Great, thanks. Nice to have those concrete examples too. A couple people have been asking about the Sap analysis. Can you explain your process for collecting samples and a little bit more detail on that, please?
Absolutely. So Sap analysis is a relatively new, well, it's a refined technology. Currently, the two labs that I'm aware of that do that are Regen Ag Labs in Oregon, which has just started doing it, and there's a company out in the Netherlands. It escapes me at the moment. I'll think about it in a second. But the basic process is you collect 100 grams of leaves, and they actually do a Sap analysis, and that is the trade secret part from the lab, as how to do that extraction. They're not testing tissue. They're actually testing sap, and it's not just the old plant sap material. The reason we do that 2-week intervals is I want to get a consistent comparison. But the other thing is, if I send a sample out on Friday, I have results on Monday. And I can look at those results and then plan ahead. The reason why we do old leaf and new leaf is you can tell that if a plant is deficient in a material, it's going to tend to take any mobile nutrient that it can from the old leaves and move it up to the new leaves. And so it's a way of predicting where your nutrient deficiencies are going to be. Also, if you have a history of it, I just know that I have iron, boron, copper deficiencies early in the season. And so it's a way of anticipating those. The lab's name in the Netherlands is called Nova Crop Health. The company that I do that with, which I would highly recommend as a very competent agricultural consulting company, is John Kemp's company. With this, I will get to that in a minute. Oh, I'll think.
You can type it in the chat later.
Yeah, I'll type it in the chat. It's just it's eating at the moment.
Yeah, Greg, this is Keith. Can you just try real quick to see if you can turn your video on?
Can I do what?
Can you try turning your video on for 1 second?
I thought my video was on.
Okay, yeah, for some reason it's coming up blank. So carry on, all's good.
Oh, the company's name is Advancing Eco Agriculture.
Great.
Katie, can you see my video, or is it blank for everyone?
It is blank, but I think it's fine. I know there's so many questions. I think that your voice is coming in loud and clear. So here's a question about fertilization and what is your approach beyond your traditional methods of applying NPK, or what are some of the other ones that you end up nutrients you have to address? How do you address that?
So I use the plant sap analysis as an indication of where I am having imbalances and issues. The first thing I would say about fertility in a fertility program is the very first thing you should stop doing is adding too much of things just because you think you should be adding them. Nitrogen's the classic one. Grapes use very little nitrogen. I also focus my fertility program in the fall. One of the advantages of being in a warmer climate is I get anywhere from 2 to 3 months of photosynthetic capacity after I've harvested. And after you harvest, it creates a auxin cytokine shift in the plant, and the plant goes kind of into photosynthetic efficiency and storage mode. So I usually do an extra foliar and a couple of fertigations That's when I'm priming my soil to get the soil microbiology going. And that's I push a lot more of my efforts into the fall, because by the time you get bud bursts in the spring, it's a little late and it's hard to get nutrients into a vine early in the season. I use two primary methods. One is foliar for immediate uptake to correct short-term deficiencies. I am leaning much more towards fertigation, long-term slow fertigation, usually mixed with organic compounds like humic acid, to try and prime that pump. I think that's an important concept with regenerative ag, is the foliars are meant to prime the photosynthetic capacity of the vine and your cover crop, so it has excess sugar to pump into the biological system that then turns on the rhizome cycle that then makes the nutrients available. That reference on rhizophagy is James White, I believe. Some fascinating work. I'd highly recommend doing a Google scholar, James White, rhizophagy. Getting a nice good bottle of wine. That is definitely a full bottle of wine research project.
Great, we'll make sure to get that in the chat too. So someone's wondering about that. There's so many different ways that you can analyze the soil microbial community. And do you have recommendations for what you think is the most beneficial?
Yeah, there's a couple. I mean, first and foremost is a shovel. It's just go out and dig, smell, look, feel, get the texture. There's a couple of cool NRCS videos. Ray Archuleta, I believe is his last name, who just does these cool things about soil infiltration and soil structure. Super easy tests that you can just do with a jar and a mesh. I am leaning much more towards the Haney soil analysis to do the respiration. For years, I was looking under a microscope. I studied with Elaine Ingham for a few years. I love her concepts. It's just counting bacteria and counting fungi were just, it's slow. It was a real challenge to do. I still do it occasionally. I'll get a sample. What I'm looking for is how prolific the bacteria is and how prolific the fungi is. So those are kind of my main indicators. But one of the reasons I was talking about, once you get the system going and you're doing less sort of emergency, corrective issues, you have more time to spend in your vineyard. And let's face it, that's why a lot of us got into this. It's fun to be out in the vineyard and it's fun to see those changes. Last year, we got 6 inches of rain in 24 hours. And I was concerned that we would have some flooding in the vineyard. And so I went out to make sure the culverts were clear, and they're actually dry. In those areas that we were developing good soil structures, it just absorbed all of it. And my neighbor's vineyard, who still sprays and discs 4 times a year and does the whole thing, there was literally vines running into the creek. When you see those kinds of differences, that's when you know you're on the right track.
Sure. Yeah, it looks like we might have time for 1 or 2 more questions. There's a couple people.
Yeah, I did. I saw one question quick in the chat that I wanted to just, someone asked about the cost of IrriWatch. It's $6 per acre per year. It's super cheap.
You said
$6 per acre per year. Yeah.
Wow, that is not much. A couple people have asked about the reduction in fungicide use. And
yes.
How did that happen for you? And can you explain that a bit more?
So 2 things. As the plants move up that plant health pyramid, they're just developing more of a natural resistance to fungal infection. The second is I spend much more time in canopy management. We hand prune everything now because I am so focused on good canopy management, good airflow. I want the fruit in that fruit zone. When I get that expression of the 2 primary apical buds, I get those 2 large clusters right in the fruit zone, and I have a large canopy over top of it protecting it. The other thing is I use an IPM approach, whereas I am looking at the Gubler powdery mildew index. It doesn't tell you everything, but it's a decent predictive tool. And once again, the most important tool I have is I know where my hot spots are. I go out and I start to look, and when you see those early indications or you see the powdery mildew index go up, that's the time to go out and take corrective action, not on a regular schedule.
Let's take one more question and then move to break.
Thanks, Keith. I guess just to wrap it up, someone's asking, I think this would be good for everyone. What do you think is one of the most important things you're doing? And would that be something you'd recommend for people who are trying to transition into more of a regenerative system?
I think the most important thing I'm doing is thinking very differently. I am no longer thinking of my vineyard as a mechanical system, but I'm thinking of it as a biological system. Whenever I have something like powdery mildew, or I have mealybugs or anything like that, I don't see it as an invasion. I see it as nature's way of being sort of garbage collectors. Is I have a problem with my system, the garbage collectors are coming in to try and clean it up. So instead of fighting the garbage collectors, I ask, what's that underlying problem? Why doesn't this plant have its own natural abilities to fend off powdery mildew or mealybugs or whatever it is? A specific example of the mealybugs is I started using a soil treatment that has a lot of chitosan in it, crab shit. And unbeknownst to me, it increased the chitosan bacteria in the soil, and mealybugs go through a nymph stage where their outer shells are made of chitosan. So the bacteria was eating the mealybug larva in the soil before they even emerged. Totally unexpected, but it worked great. And it's just a great example of how those kind of systems kick in.
That's great. Thanks so much for sharing. And sorry we didn't get to all the questions, but please reach out to Greg or again, any of your local RCDs to hopefully try and get more of these answered.
Yeah,
Thanks
I'd be
again.
more than happy. If anyone wants to reach out, it's greg@wilsoncreekwinery.com. Would love to hear from folks. Thanks so much. Also, if you reach out, let me know where your winery is, because I love hanging out with other winemakers and tasting your wines.
Great.
Thanks,
Let me
everyone.
jump in there and say thank you so much. That was excellent, Greg. And we probably could do another hour of question and answer. So maybe we can get you up here or virtually get you up here again. That was excellent. Thank you. And thank you to both the presenters. So far, it's been great. We're going to take a break. We're slated to come back at 10:20 and hear about climatic water deficits, trends, and implications from Lisa Micheli. And so we got just under 10 minutes, and then stay close to your computer and we'll get going soon. Thank you.
Great. Well, hi, everyone. Thanks for having me today. I had a panic because I just saw that Symposium was misspelled on my first slide, which is previously hanging out under one of my logo leaves. I'm really delighted to be here. My background is actually in fluvial geomorphology. So I'm really stepping out of my expertise here, but I work in a generalist way, thinking about climate trends on our water cycle, and how that impacts our ecosystems and how it can be applied to agriculture. So I'm looking forward to chatting with you today. And if you don't remember anything else from my lecture, the take-home message really is our region is getting more arid. I'm going to give a bunch of examples today from Sonoma County, but of course we're seeing similar trends. So we'll talk about that, how we're measuring it, how we can conceptualize it, and some tools that may be available, that are available that may be applicable to some of the questions you're asking yourselves in the context of agriculture. So now I just have to make sure I know how to go forward in this brave new world. I think it's the two-screen thing. Okay, so if you don't know about Pepperwood, you haven't heard about us, we were a 3,200 acre reserve that was established with the California Academy of Sciences. And in 2009, we became our own foundation and opened this center. This is a picture of the center right after it was constructed. And we've become a hub for meetings like this when we used to meet in person, and then a place where we are measuring all the different aspects of climate change in real time on our natural ecosystem. We do also support a grass-fed beef operation on site as part of our stewardship of 900 acres of grasslands. The work I'm sharing today really comes from a bunch of colleagues that were brought together through the Gordon Betty Moore Foundation in 2010. And we were asked to collaborate across boundaries. So this is a group of hydrologists, ecologists, plant specialists, coastal modelers, fog modelers, and the Moore Foundation asked us to think about what's the impact of climate looking like for the wild and working lands of the Bay Area. And so I give credit to this whole group, and you can learn a lot more about them on our website. So I'm just going to go quickly through sort of historic and projected temperature and rainfall trends for Sonoma, and introduce this idea of climatic water deficits, which many of you may be familiar with. I often call them soil water deficits. And this is a term that actually brings together the combined effects of warming and rainfall variability. And then I'll show you where you can explore more of this data on your own, and I'll be available for any questions. So I decided to tap into this website. It's called the Sonoma County Climate Resilience Dashboard. It was created as a prototype for the National Partnership for Resilience and Enhanced Preparedness. It's a site that you can revisit that summarizes results that we made for Sonoma and the Russian River. We do have similar data available now for all of California as well as the US West. So I'll talk more about that. But the take home to start with is that our climate is getting warmer and more variable in terms of rainfall. So these are two typical plots. We have the data from 1920 to about 2000, which is the historic data, and then a set of projections on the rainfall. And we don't know in our region whether we're going to, on average, get more rainfall or less rainfall. The challenging part of it is that we're actually looking at more extreme events. So when we run these models, we're seeing maybe double as many flood years as we had in the historic, previous 100 years, and 60% drought years across models. And those are very rough numbers, suggesting that basically we just have more uncertainty and probably more extreme events punctuating our precipitation record. And then down below, you're seeing the historic trends of temperature and projected temperature trends with fuzz around them. And I think it's just a different, it's an important take-home message that we know that the temperatures are increasing. The uncertainty is about how fast that is happening, but we don't know which direction the rainfall is going. And that is because in terms of accumulating emissions in our environment, we have a good model that's quite rigorous about how that affects the warming effects, but there's really just a lot of uncertainty about the mechanisms underlying rainfall. This is a slidey tool. I was not brave enough to try to do a slidey tool in the presentation, but I fast forwarded all the way to 2070, 2099. And all of this kind of climate projection work that we do is done in 30-year increments, which is sort of you can think of that as one data point for looking at long-term trends. And these are models that we've developed with the U.S. Geological Survey, which are publicly available. Where we have observed the greatest warming to date is actually in our winter temperatures. Our winter and our nighttime temperatures are warming faster than our daytime temperatures and our summer temperatures. This shows you the pattern of where certain thresholds are exceeded by end of century in terms of monthly temperature averages. And I think a take home is just that when you compare that to the daytime temperatures and the summer temperatures, it's a different pattern. So in terms of whether your crop or your ecosystem of interest is more sensitive during the summer or during the winter, you might see a different pattern in terms of the degree of warming that we're going to experience in the landscape. This is a visualization of an atmospheric river. These storms that form over the middle of the Pacific Ocean and then drench us, they can carry up to 35 times the amount of water in the Mississippi River. And they're very, very difficult to predict because they form quickly and they literally meander through the sky. And that's the source of the uncertainty, really, and how much rainfall we're going to get over the long run. We don't even really have the ability to predict the behavior of these in a quite short-term period or exactly where they're going to land. So just to keep in mind the mechanism behind the rainfall uncertainty. When we're looking at climate change and water supply, the model that I'll be introducing, we can look at both surface runoff and recharge. And one of the take homes when we're working with communities is just to realize that our groundwater recharge is relatively constant and relatively resilient with respect to these variations in rainfall. And that makes it all the more important, all the groundwater protection work that we're doing right now to protect that source. And you can use runoff that's available to fill ground on an annual basis, brings that idea home that it's our savings account. So we mentioned more frequent and severe droughts. And one of the concepts I'm going to try to bring across today is the difference between a water supply drought and a landscape drought. And we've learned a lot about this looking at the 5-year drought that started around 2011. And we're still seeing, of course, droughty years happening right now. So we use a US Geological Survey model called the Basin Characterization Model. And it basically keeps track on a grid cell basis of the impacts of temperature and the solar radiation combined with precipitation on the ground surface. And we use a model, a watershed model that has the topography, the thickness of the soil layer, and then the underlying geologic material with estimates of the permeability of that groundwater material. And it does estimate how much water goes into deep storage versus being discharged to the streams. And the stream discharge is one of the ways that we validate this model and make sure that the model stream discharge matches what's happening. At Pepperwood right now, we're actually putting in a flux tower that will directly measure evapotranspiration from the soils and the plants. And we'll talk more about why we feel like that's so important to get right. So water deficit, and I'm sure many of you are much more expert soil scientists than I am, but we estimate this term using the potential evapotranspiration, which is the amount of evaporation that would happen if you just had a bucket or a tray of water out and water was unlimited based on the temperature and the solar radiation. How much evapotranspiration would you expect? And then the nice thing about our basin characterization model is we actually keep track of soil storage over time. So at any particular moment in either history or the model, we have an estimate of how much water is in the soils. So if you think about the dry down summer season, we're depleting that stored water out of the soils, and there's a point at which you're going to run out of it. Because it is not an unlimited supply. And so it's that delta between the potential and the actual that we term the climatic water deficit. And it's an indicator of drought stress. And so I'll just say from an ecosystem point of view, this became a game changer when our hydrologists were able to estimate and map climatic water deficit. It explained why different kinds of natural vegetation in the Bay Area can occupy the positions that they do. And we were able to much better estimate with David Ackerley's lab, who's at UC Berkeley. He's now the Dean of the College of Natural Resources, to much better estimate where plants can grow. Because obviously in a water limited environment, your tolerance for different levels of deficit will open up different landscapes. And a lot of it has to do with how much water can be stored in the soil, and in particular soil depth as a driver of that. But when we fast forward the models, and we have worked with 100 different models, I'm using one or two just to show, but we're looking at perhaps a 10 to 20% drier situation by 2100, where the water deficits add up to about 3 to 6 inches of incoming rainfall. So that's a pretty big difference to make up. And again, this is a more extreme model. We could have milder effects, but it sort of gives you the range of how much liquid water that used to be in the soil will more typically already be in the atmosphere. One of the things I like maps, is we can map water deficits across the landscapes. These are some of the early estimates we did. But the other thing to think about is that these inland areas, and this is actually Pepperwood, our property over here, yes, they're subject to a lot of water deficit change, but they also are relatively high water deficit areas to start with. So we started thinking about, you know, perhaps there's more vulnerability in the coastal areas, which normally don't see much change in climatic water deficit, that maybe small changes there could have big impacts. The other thing is when you start doing the math about water deficits, on the left-hand side, these are all the different basins: Marin, Petaluma, Sonoma, Napa, different places where I, before coming to Pepperwood, was particularly working on stream restoration type projects. So we wanted to differentiate between the different valleys. But then when you plot water deficits, especially in the second half of the century, the impacts in water deficit are accelerating faster than temperature. So that seemed like an important take-home message to bring. We also started working on visualizations like this that show you the trade-offs in different elements of the water balance over time. On the left-hand side, we're looking at our home watershed, Mark West Springs watershed, from the average from 51 to 80. And we've got the actual evapotranspiration in green, runoff in dark blue. That's the medium blue, recharge in dark blue, and the accumulation of soil storage over the season. And then you can see this dry down period. And we started plotting the water deficit as it accumulated during the dry season, which gives you a visual of how in a warmer future environment, we're going to see those deficits grow. And there is a link to this particular tool. You can download any sub-watershed in actually the entire Bay Area and have a chance to see the historical and projected numbers yourself, look at the monthly values, etc. So when I talk about different kinds of drought, and this really was a big take home during our most recent drought, you know, we often think about the drought is a drought when the reservoirs are low. And reservoirs generally capture surface runoff and shallow recharge. And that's super important because of folks who rely on those reservoirs for water supply. But then the landscape droughts, which are really the drought effect that climatic water deficit is measuring, what we started to realize is that in a super droughty situation, you could actually be accumulating deficits from year to year. So if the soil dries out extensively in a particular year, and we only get, say, 7 inches of rainfall, which is where we were at Pepperwood last week, you might actually never completely replenish the soils and get them back to a zero deficit condition. So this was an important thing that we realized could help differentiate people's understanding of drought and really some of the lasting impacts of drought that can occur on agricultural and forested lands that we hadn't necessarily accounted for. The other thing we're showing in this little visualization is 1977 and 2014 had relatively similar rainfall totals, but the amount of warming that we've had just in that small window, which is only about 1 degree Celsius really on these average temperatures, created a drier condition for our region. So same amount of rainfall, but drier condition because of the warming trends that we're seeing. I also thought this was a neat visualization. This is, again, looking at one water year, 2013-14. And on the left-hand side is what we would generate for, say, the water agency to understand the difference in terms of water supply relative to our pre-change average. But then on the right-hand side, then we're mapping what is the water deficit condition and how dry are these soils. So 2 different metrics of drought that we could be thinking about. I mentioned this idea of where climatic water deficit exceeds historical variability. This is something we can map. This is taking 1 standard deviation as the threshold. So anything gray is staying within 1 standard deviation of change. And this is a mid-century versus end of century comparison. And it was interesting because when we first started working down here in the southern part of our region, this is the North Coast region, I should clarify, going from Sonoma County all up to the Oregon border, we saw this sensitivity along the coastal areas. And again, they are going to go outside of their historic range faster. than the interior. However, as you get further north in our region, that is a flip-flop situation. So you can see there's some value to using these high resolution climatic projections to understand the variability of the landscape. So once we get this water balance model going, and it is, I said, publicly available to US Geological Survey, there's other applications we do in terms of our ecosystem work. We can model river discharge. We can look at vegetation exposure. There's actually a statewide California report that one of our team members did for Fish and Wildlife, looking at vegetation exposure. And then we can also use CWD ends up being one of the major predictors of fire hazard. Because we're doing some work right now about how the climatic water deficits in the soils correlate to the live fuel moisture and the degree of desiccation in the leaves of the vegetation. Just some other proof of concept we did when we were working with Sonoma Water, for example, taking historical records of irrigation demand out of the Russian River and projecting that for future climate to understand how irrigation demand may increase. So we think that that's pretty directly related to water deficit estimates. I mentioned the ability to model where natural vegetation will be able to persist in the future. And of course, that is a really different picture, whether we end up in a wetter scenario or a drier scenario. Personally, I have a feeling we're going towards the drier scenario, so we can think about the changes in natural cover. And again, the fire models, we came out with these fire models in 2016, right before almost everything in heavy red on the right-hand side of the map burned. And then, of course, last year, we started catching up on some of these dark red areas on the other side.
Lisa?
Yes.
Yeah, you're doing great. You got 10 minutes for your presentation, then question and answer.
Okay, great. Just letting you know that we are working a lot on habitat corridors and land connectivity. And it's definitely the mosaic of agricultural and working lands that enable us to conserve landscapes and create basically climate adaptation pathways for some of the wildlife we're worried about in the landscape. But I see CWD, the drought hazard, and also the fire hazard as an overlay that we need to put on these kinds of concepts about habitat connectivity and seeing the functionality of ag lands to buffer some of our areas, particularly with respect to fire. And some of our team members have done work specifically for grape varietals, coordinating water deficit and temperatures with historical grape ripening dates and starting to look at the suitability. And I think that this is a great example of how the climate gradient in our area at the sort of county or regional scale is really a east to west gradient. So, I think people typically think about climate to cool off. You go up the mountain or you head north. But here in our region, we actually head towards the coast. And what this map showed us is that there's going to be a lot more areas that will be suitable for grapes towards the coast in the future. That's the trend that we're on. And that there may be areas in the interior that start to become perhaps too hot, at least for the varietals that are growing there. And I know it's a big topic of inquiry within the vintners community right now. And I have vintner friends who are testing different varietals, say in the Napa Valley, to see what's going to be robust in the climates to come. So I am a real believer in the fact that everything I just showed you in terms of models in the future are hypotheses. At Pepperwood, we have 17 soil moisture probes actually installed as of right now. We are really interested in this question of the water balance in our rangeland soils, which really are classically similar to most agricultural soils in the area. One of the things we discovered, for example, is that this black dashed line had been considered before our big drought, 5-year drought, the lowest that water potential could go in the soils. And the benefit of actually measuring it was to realize in these new hyper drought conditions that the soils can get drier than we thought was physically possible. And we have now been able to adjust the models to reflect that reality. And this is just showing you the data that we've been continuing to collect really trying to drill down into the impact of individual precipitation events on the soil moisture. So these little spikes are rainfall events that we're capturing at our weather stations. And then each of these lines is actually a different soil moisture probe at variable depths. And so we're really interested in analyzing what we're calling these sort of dry down periods, looking at the influence of antecedent soil moisture, the soil moisture that's already in the ground, on the ability of the soils to serve as a reservoir over time. And then looking at those very dry conditions, how quickly do things drop down? How much does it, depend on the rainfall that we've gotten? And how much does this vary across the landscape? And we just submitted, as I mentioned, the connection to live fuel moisture, a proposal to be measuring live fuel moisture and vegetation water potential in our natural vegetation types during this period, so we can tighten up that relationship. Another area of TBC3 study is the frequency of fog. Fog is actually a very unknown factor in terms of how it might change over time with climate change. There are some models that think we'll get more. There's some models that think we get less, but certainly fog is our natural air conditioning. It can reduce climatic water deficit in soils by both, impeding or preventing or reducing the incoming solar radiation hitting a patch of soil. But then also the fog can be a direct water source to redwoods and others that know how to harvest the fog. And that's something that we're measuring. We have a station at Bodega Bay and we have a station inland at Pepperwood looking at the actual liquid water content in our fog and tracking that over time. One of the tools I like, if you are not a mathy person and you just want to say, hey, what's the bottom line in terms of what Lisa is saying about the future of our region? We did an analysis with Sam Veloz at Point Blue where we looked at the current landscape and we classified it in terms of the winter minimum temperature, the summer maximum temperature, water deficit, and precipitation. And the areas that are most like us in the future are really San Bernardino County, Santa Barbara County, these red dots down here under the drier and hotter rainfall. We will be looking at conditions more similar to this area. So, we might want to be thinking about in the long term, how do we gracefully adapt to a transition to this drier environment? How do we increase the water holding capacity of our soils and work on our water security? These are the different tools I mentioned. The Climate Smart Watershed Analyst has monthly time series going back through time and forward through time. For 18 scenarios, there are 100 scenarios for the globe. These are the 20 we felt provided a full set of variability or possible conditions for our area. So you can click on your watershed and get reports. And all of our work is really at this climatecommons.site. If you want to play with the dashboard that I showed, it's here. And if you want to download all of the BCM data, it's also available through the Climate Commons. Here's the landing page for the Bay Area tool. And here's an example of how you can turn on and off these different time steps. And this is the tool that had that seasonal plot of CWD to tap into. And then there's a lot of data that's made available through CalAdapt. It tends to be a larger scale. Many data sets, but it gives you a chance if you want to look outside this region to find resources. So that's it. The take home is really, we're looking at more arid conditions. How do we work together to manage our resources under that situation? And with that, I'm happy to take questions and comments.
Great. All right. Thank you very much, Lisa. You came in a little ahead of schedule and great presentation. As someone who could talk about the weather all day long, that gave me a whole lot to think about, and I have a whole lot of questions. I do encourage people to put your questions in the question and answer box at the bottom right. And then Katie, you will be leading the question and answer session, so take it away.
Thanks so much, Dr. Micheli. We do have a few coming in. One is, does Pepperwood measure rangeland evapotranspiration via surface renewal or other methods?
So the only direct measures of evapotranspiration are these flux towers I just mentioned, and they're actually both in forested watersheds. So one is a thinned watershed and one is a non-thinned watershed. So we're really trying to look at the comparison of that. We do have, as I said, 17 weather stations primarily in our rangelands, and that's where we can do really high quality estimates of the evapotranspiration, but we're not measuring it directly, but we have all the ingredients to use the Penman model to make a good estimate.
Great, thanks. Could you talk a bit more? Oh, I'm sorry. Did you have more to add to that?
No.
Oh, okay. Could you talk a bit more about the connectivity in the wildlife corridors and where someone might look for more information or existing models on that?
I love this question. So we just published 2 articles in a journal called Land. And if I can figure out how to do it, I will put the links in this chat box to the journal Land, and it's called the Mayacamas to Berryessa Connectivity Network. All of that data that's relevant is also on the BIOS database of California Department of Fish and Wildlife. So if you have any trouble accessing that, please do just contact me with this contact information and I'll make sure you can get it. We're really proud of it. And we really are, I think, the only ones who've measured sort of the climate adaptation value of the connectivity. So we're proud of that. And then we also talk about how we worked with stakeholders to come to design these habitat corridors. So.
Excellent, thank you. How does the CWD look into this spring versus like the last spring?
This is where I wish I had time to do some math for you, but I can guarantee you that this spring is drier right now. As I mentioned, we only had 7 inches of rainfall as of last week. I'm so happy that we've gotten these storms coming in right now, but we're very concerned about the extent of drought this year. It seems much more extreme than last year, but if we have a miracle March, that could really do a lot of good. And the timing of rainfall is really important in terms of that end of season climatic water deficit, because if you do get your rainfall for the same amount of rainfall, if you get more of it later in the season, you will see lower deficits in the fall. So that's a great question to raise. I see the question from Jack about relating to aquifer recharge. Jack, there should be a basin characterization model for you anywhere. If you are in the Bay Area, I want you to use that tool that I just showed you. But basically, we can make a map of recharge potential, and the units are millimeters per year. So it's an estimate of recharge potential. It doesn't necessarily quantify exactly what's going to get into your aquifer. It's quantifying what's recharging below the soil layer into the weathered bedrock. So you generally, if you're really wanting to be very precise about the amount of water in the aquifer, you need to add what they call a GS flow model to it. And that is how, at least in this region, most of the groundwater plans are using the basin characterization model to estimate that surface recharge and then coupling it with a GS flow model below. But if you have your property, we have like a 30-meter layer that would enable you to see the variability of groundwater recharge potential across your property. Do you want to take the next one, Katie? Because I'm probably losing them.
Let me ask this one. Is measuring water table depth instructive regarding climate water deficit? And would you say there's a correlation there?
No, I would say they're not necessarily correlated. So the water table depth will give you a good estimate of where the standing water is, irregardless of the soil bedrock transition. The water deficit is really driven by what's happening in that top layer of soil, regardless of what's happening underneath it. So I don't think the soil sees the water table per se in terms of water deficit. However, the soil, of course, is what's delivering the water to the water table. So I'd say it almost goes the other way around, that in high deficit years, you'd probably be getting less throughput to the water table. But it by itself doesn't necessarily drive what's happening above it.
Great, thanks. And this is stating this as a follow-up question, but are your weather stations and Penman stations compatible with the SEMIS protocol station setups? Again, pasture grazing settings, and are they clear of trees and wind turbulence? It's kind of a complex question, but
yeah. I believe so. I don't know all the SEMIS protocols exactly, but I can definitely answer this question if I talk to our tech. But I believe that we're meeting all the highest standards in terms of open territories for the stations not having weird wind effects that would mess up the anemometers. We definitely mow around all our stations, and since we do have active cattle grazing, they're also fenced. So they're fenced, cleared, mowed, etc. In some cases, we put weather stations under the trees because we want to see what the impact of the trees is on them, but everywhere else where we're looking for straight weather data, we're definitely out in the open.
Great. I'm going to switch gears a bit to a little bit of a broader question, but could you summarize maybe your top 3 to 5 takeaways for wine growers based on all this climatic data and the predicted changes, and how might they adapt based on this?
Well, my first take home is, as outside of the wine industry, a benefit for everyone in the community is to understand that a lot of our grape growing operations are on important recharge areas, particularly alluvial fans that are well-drained soils where you see the creeks dry out. And personally, I feel like this is a great win-win solution because we have a valuable crop and it's also a relatively low chemical use crop, and it's a great way to keep our aquifers protected and our recharge protected. And I often counsel people that if you're anti-vineyard, the next thing that's going to come in is a housing development, and that is not going to be as functional for our aquifers. I have a question for you all, which is this real emphasis lately on the idea of carbon additions and the ability for carbon additions through either composting. Of course, we have a major forest thinning product problem happening where there's a lot of basically slash that needs to come out of the forest. You know, are there potentials, are there opportunities to augment the soils with carbon and increase the water holding capacity? That was the subject of a big analysis for the 4th climate assessment for California that the BCM modeler sort of took on, which is where are the soils thick enough where you could have an impact if you could add carbon. And if that's something that's a win-win, again, for the growers, it's also a win-win, I think, for our community because you're keeping more water in storage in the watershed for release later. And then, you know, I think about having worked, before I came to Pepperwood, I worked in the Rutherford Reach with growers on a major restoration project for the Napa River. You know, what are the things you can do on site? And I know there's a lot of experiments happening with orientation of vines and pruning strategies to keep your grapes at the right temperature and to protect against this incoming solar radiation. And I also think the question is, are there varietal changes that you might want to be considering? And I know, for example, at Larkmead, since they're not going to move to another property, right? I think you have 2 different situations. If you're the farmer who's staying on your farm, you're going to adapt there. I know that some of the bigger companies already are looking at their portfolio, like should they have more lands further north to be able to adapt to the situation. So those are sort of my 3 biggies, I guess.
That's excellent, thank you. I will say someone's posed this response to one of your questions, and it's something that popped up too, is this big interest in biochar. And I know that people are starting to study that more in our region. So that could be an interesting thing to continue to study to see if it makes sense in our viticulture systems.
Yeah, I think biochar is great. I think the challenges with these things are like, how do we get the material to the biochar unit and then get it back to where it needs to be without having a bigger carbon footprint than we started with?
Absolutely.
And so I'm really interested. I know that there's portable biochar units. That might be a great a great option. But yes, we're curious about that. And we have tried, you know, because we can't haul out all the materials in our forest, we are trying to do biochar style burns, which are lower temperature burns in our burn pile. So we get as much of that charcoal type product as possible.
Great. Here's another question. Could you compare the evapotranspiration use for plants in vineyards versus maybe rangelands versus forests? That's a pretty big question, but
I love this question. I can't do it on the spot, but I know how to do it. So now, now I'm going to take that as a homework assignment to look at comparative ET, and I will get it back to Keith so he can send it to the groups. But let me just make sure I have it right. Forest, rangeland, and vines, right?
yes.
I love
If you have
that.
other systems you'd like to throw in, go for it. But
yes, I'll take on that challenge.
Awesome. Thank you. I'm sure people will look forward to that. So just getting this one in, can you discuss the evaluative rubrics or assessment criteria that you utilize to understand trends in aridity with respect to wildfire risk and in relation to landscape types present at Pepperwood, for example?
Yeah, so this is one of the exciting areas of interdisciplinary research where we started modeling this water deficit at a high term, you know, high resolution, which is really great, right? Because I'm a physical scientist. So if you can physically model something that matters in ecosystems, it's a lot easier than measuring the ecosystem. So we are taking live fuel moisture, which is, you know, basically trimming the plants and then looking at the total percent water content in the plants. And that's a very laborious process that can only be done manually at this point. So we're finding that the climatic water deficit is a great predictor of live fuel moisture. It's lagged though in time, right? So it depletes first and then the live fuel moisture depletes. So we've just applied to the Department of Energy actually to scale up that assessment and look at more habitat types, kind of do a coast to crest transect in California where we're making those measurements every week to really get that relationship firmed up. And I will say you know, this was inspired by the fact that after the 2017 fires hit our region, we had the opportunity with NASA to do an analysis. We had a very high resolution LiDAR derived veg model, 5-meter veg model. And we looked at all the different parameters that were correlated to canopy damage. So assuming that where the canopy was lost was some of the most severe fire impacts that we had. And the number 1 and number 2 items were the density of ladder fuels, which we estimated using the LiDAR data set, querying the understory, which is not a typical measurement. I think we're the first ones to really try to assess that data. But the density of ladder fuels between 1 and 4 meters, and then the climatic water deficit the month the fires hit, were the 2 best predictors of fire severity. So it's now folded into the CAL FIRE. One of our TBC3 partners is the author of the model that CAL FIRE is looking at in terms of looking at the impacts of climate on fire hazard, and those changes in water deficits seem to be a really important predictor of where hazard's going to increase.
Very nice to hear that all that research is getting on the ground.
Well, that's what we try to do, is like we try to do very applied work that answers questions people have right now. So we tap into those great university and ivory tower researchers, but only the ones who want to work on real problems that we're facing today.
Of course.
Yeah.
Well, we only have a couple more minutes, and I know Keith actually has a question that he might like to ask you. If you want to jump in there, Keith.
Well, I have lots of questions, but I'm going to stick with one right now. I remember when you presented 8 or 9 years ago, something that really struck with me is, and I know this is just modeling and there's no way to know reality, but you mentioned that like things seem to appear that perhaps around the year, if I have it right, 2070, that our region would look a lot like Santa Barbara.
Mm-hmm.
So that was 10 years ago. So that's more than 10% of the time in between. What would you say the pace of things are as they're actually playing out? And then what's, you know, how fast do you see this sort of drying out or this hydrological change happening now?
Well, I want to put like quotation marks, when we say 2070, we're really like saying 2070 to 2099, because again, it's 30 years of a data point. So we have now traveled, since you first heard my presentation, only 30% through one data point. But without having all the math in front of me qualitatively, I mean, when we went into the drought, the 5-year drought around that ended around 2015, and going into these fire seasons, I mean, we are seeing what we estimated as end of century impacts. And that's what's been kind of hard about this work, is it's not a gradual transition that's happening. We kind of went right into this extremely dry, hot, and difficult weather. So I'd say we're moving faster than we thought, but I can't tell you that's a trend, Keith, because of the variability and the uncertainty in our weather systems. I mean, maybe if we have another meeting 10 years from now, it's going to be like this incredible Arctic vortex that pushed a bunch of rain and cold weather on us that, they're having in Kentucky right now. So, so I'm leery of saying that we're on any particular trajectory. And that's one of the things that people always want to say, well, which one of the models is turning out to be right? But we're always like, you know what, those models are just models, and they represent everything that physically could happen. We're going to keep measuring and then be able to tell you more accurately what really did happen. But I would say, compared to when I first gave this talk, I thought that these things we've been experiencing the last few years were further away in our future. And I tell people, I wish our models hadn't been right. Like, usually scientists want to be right, but in this case, it would be nice to have been more wrong.
Right. That makes perfect sense. And I think really what's getting people thinking about that is these yearly fires that we're having now, that seem to be further down the road. And really, I've lived here almost 20 years with seeing almost hardly any fires, or none of significance, and now they're a yearly occurrence. So I think that has everyone wondering, how fast this is happening. But that was an excellent presentation. I think really helped people who are attending to see where the trends are and where they're going. And I thank you very much for that. And we'll need to talk on our own soon, because we are looking at some of these very same questions of where we're working on a lot of fuel thinning to respond to the fire. And then I'm also talking with growers who are looking at applying mulch, and both in rangelands and vineyard settings. And I see a potential there to really address two things at once and have a more positive outcome. So
I think the more we can work together, the better. And we're all one living system. It's a mosaic of working in wildlands. And I really appreciate everyone taking the time to listen to my presentation today. Thank you. And come visit us at Pepperwood. Just contact me, come up for a tour.
Yes, thank you. Yeah, if you haven't been to Pepperwood, it is an amazing and beautiful place.
Anya's asking me, I'm going to put some links in the chat box for some of the data, Keith. So
yeah, and if
you can harvest it there.
You could put a link to Pepperwood too, if someone already hasn't.
Okay, thank you.
All right, thank you, Lisa. That was excellent and fit in very well with everything we've been discussing today. We have one more section of our event today, and this one's even closer to home because this is part of what we call the North Coast Soil Hub. This is a North Coast Soil Hub production, I would say, which, to remind folks or those who aren't on, there are several people involved with that, or several organizations, but it is led by the RCD, and it's the Napa RCD, the Mendocino RCD, the Sonoma RCD, and Ridge RCD. So those three counties are all involved, but we will be expanding, and I know Lake and Marin are coming on, and we will have a new coordinator soon. And so we look forward to continuing efforts like this. There's a lot more though, just than these events. And so we're going to spend the next section talking about what the North Coast Soil Hub is up to. And we're very fortunate to have Miguel Garcia from the Napa RCD, who has been doing a great job leading the project and leading the folks from different RCDs, and also coordinating. We NRCS and other organizations have been participating too. So he's been coordinating that, and he's going to talk to us about it. He is the Sustainable Ag Program person over at Napa RCD, and he has a very similar job to myself as the soil and water specialist. And we're also going to hear from Shannon Cappellazzi from the Soil Health Institute, and she is helping us with the project, and will tell you about what she's up to, but it's great to hear from her because it was about 5 years ago when I first contacted her about working with the Oregon State University lab where she used to work. And this discussion began, and then she's taking it much further, and now she's with the Soil Health Institute, and it's going to be great to hear from her. So with that, Miguel, the floor is yours.
Thank you so much, Keith. Can you guys see my presentation just fine?
Yes, you're all good.
Okay, perfect. Thank you so much. Today, I will talk to you guys about what the North Coast Soil Health Hub has been up to with regards to our soil health assessments and field trial monitoring in vineyards. And I'll give you a little bit of background about the hub. The North Coast Soil Health Hub is a collaboration between the RCDs in Sonoma, Mendocino, and Napa counties, and more recently, we're working on incorporating also Marin and Lake counties. For the past 3-4 years, we have been focusing primarily on vineyard systems, but by bringing Lake and Marin counties, we're trying to expand on the kind of work that we're doing so that we're looking at soils in other agricultural systems other than vineyards. And this is an effort that we've been doing with the Natural Resource Conservation Service, the NRCS, which is a branch of the USDA. And also we have been collaborating with the Soil Health Lab at Oregon State University. Today, there's a greater recognition that soil health plays a key role in minimizing the impacts to water quality and increasing drought resiliency and other impacts on climate changes like we heard from our previous speakers. So the North Coast Soil Health Hub was created as means to raise awareness about these issues among local farmers and to communicate with them about the benefits of soil health for crop production, and also as a strategy to address the region's resource concerns. The hub serves as an active learning network for farmers and agricultural professionals who share information about soil health practices. We have our website, soilhub.org, where we try to communicate about all of these efforts, and this serves as our communication platform where information about ongoing field trials and regional specific soil health information is shared. The hub is intended at some point to be part of a statewide farm demonstration network, which is currently under development by the state, federal, university, other resource conservation districts, farm bureaus. So eventually we would like this whole hub to be part of this, be linked to this network so that the information that we have can be shared and can be discussed with other farmers who have similar cropping systems throughout the state. So the North Coast is well known about the viticulture work, but there's other regions in California and other parts of the country that could benefit from the work that we do. So we encourage you to go to our website, soilhub.org, to learn more about the work that we're doing. And if you have any questions, you can reach out to us there as well. This is the centralized location, so if you're looking for contacts on any specific RCD, this is the place that you can find us all. So as I mentioned, the main goal of the hub, or one of the main goals of the hub, is to increase the adoption of agricultural management practices that help maintain and improve soil health. So to do this, we have focused on two main areas. One of them is education, and the other one is research. The hub has made many efforts to facilitate the transfer of information regarding soil health to North Coast farmers. And one way is by hosting educational workshops such as this one. Like Keith mentioned, this is fourth year annual soil health symposium for Sonoma County. All the other RCDs, we have also been putting some soil health related workshops throughout the years. This is the first year that we do it together as a hub because we recognize that we are pretty much trying to communicate the same message. So we hope this is one of many years to come where the Soil Hub comes together to do these kind of workshops. In the future, we hope that we can do them in person so that we can do a little bit of hands-on learning and show you guys some of the work that we've been doing out in the field. The other way is by disseminating information related to soil health through our website and e-newsletters. So I encourage you to sign up to our e-newsletter if you haven't done so already, because that is the best way that you can learn about our ongoing research and upcoming events. In regards to the soil health research, we have been doing two primary things. One of them is the establishment of experimental trials to assess the long-term soil health and economic benefits of implementing various sustainable farming practices. And I will talk in detail about this in a minute. The other efforts that we've been doing is conducting soil health assessments in various vineyards across the 3 counties, Mendocino, Napa, and Sonoma. And we have been doing this to determine how different soil management practices impact soil health. So I will go over a little bit of the efforts that we're doing, and the next speaker, Shannon Cappellazzi, after me, would give a little bit more information on how we're utilizing this data. So looking at our experimental trial sites, these were established in order to assess the long-term soil health and economic benefits of implementing very sustainable farming practices. And this is important because we want to do this in a region in order to be able to figure out what works and what doesn't work in our specific region. We want to make sure that the concepts that we're trying to communicate about soil health are implemented in a way that would, as accurately as possible, represent what's going on in a region, so that we can represent as accurately as we can our different microclimates, soil types, and topography. We have collected soil samples at the beginning of all these trials. Some of them have been going on for 3 years. Some of them have been going on for a little longer than that. Some of them for a little less than that. But we collect soil samples at the beginning to create a benchmark. And we have either already collected soil samples this year, or we will be doing it soon to compare from the benchmark. And this will allow us to see what it's improving in the soil, what's working, what's not working. We intend to continue these experimental trials for a few years and continue tracking the progress. We should have more information to share with you about these trial sites and the data that we're collecting in the incoming months. So most likely you'll hear a lot more details about the actual data for these trial sites on our next Soil Health Symposium next year. So stay tuned for that. And we hope that we will be able to do it in person at that point, so that maybe we can have field days in some of our trial sites so that you can see firsthand what is going on there. In the meantime, if you're curious about more details about these specific trials, you can check them out in our website, the Soil Hub website. So the first trial site that I will discuss is the Wichita Creek Demonstration Vineyard, which is in Napa County and is owned and operated by the Napa County Resource Conservation District. This is located in the Carneros AVA. We have a carbon pump plan in place at Wichita Creek, which is assessing the long-term benefits of compost, biochar, till versus no till, and sheep grazing. In this vineyard, we have vine rows that have not been tilled since the late 90s, and they're right next to rows that are tilled every year. So this serves as an excellent side-to-side comparison of the long-term impacts of intense tilling. And it's important to do it side-to-side so that there's no soil or topography or microclimate differences. We're also testing how compost and biochar benefit soil health. And here we have plots that have either received compost or biochar, or both, so that we can assess the cumulative effect of biochar and compost, and then we can see what they do on their own. We also partnered up with Dr. Emily Gardine from UC Davis to assess how sheep grazing benefits soil health. We have been grazing sheep in our vineyard, one block of our vineyard, for 3 years now. And a graduate student from UC Davis working with Dr. Emily Gardine is assessing changes in soil health by having the sheep there. And they're also doing some assessments on how the grazing is displacing weeds. So very interesting stuff. We expect to have results to share about all of this by next year. The next trial site taking place in Napa is Gamble Ranch, which is located within the Oakville AVA in the middle of Napa Valley. And the vineyard is, the property is 130 acres in size, and about 525 are planted to vineyards. Here we are trying to understand how different compost application methods can affect nutrient and soil organic matter accumulation. And here we're looking at just what happens when you just do an overall application of compost without incorporation, And then we'll compare that with once you do an incorporation, what kind of differences we see there. We're also assessing how different tilling techniques impact soil health. So one of the things that we are assessing is the minority plow, and forgive me if I'm not pronouncing it correctly. So we're trying to see how this plow technique may affect soil health. In Sonoma County, we're doing a trial site at Schoen Farm, which is owned and operated by Santa Rosa Community College. In here, we are testing the establishment of different cover crop species. Some of them are Blando Brome, Crimson Clover, Rose Clover, Sorrel Fescue. This vineyard also has a carbon farm plan that includes compost applications and reduced tillage. So these are long-term plans that will, in a way, change a little bit of the management. And this allows us to track over time carbon sequestration related to different management practices. Also in Sonoma, we have a trial going on at one of Jackson family vineyards. And in here, we are comparing how the frequency of tilling influences soil health to wine productivity and soil carbon sequestration. So we, in the previous trials, as we're looking at till versus no till, and then we're looking at different types of tilling with the minority plow. In this trial side, we're looking at the frequency. So how often you do it, how does that affect the incorporation of carbon into the soil and other soil health indicators and vine productivity, and how this affects also the compost benefits over time. In Mendocino, we have Farrington Vineyard. And in here, we are testing the effects of several common cover crops and tillage management on soil health and vineyard growth. We have a couple different experimental trials. One is a no-till with a receding annual cover crop. The other one is a till with a biomass and a green manure cover crop. And then an alternate rows till system with the first two cover crop regimes, and also a no-till with a naturalized vegetation. More recently, this year, we're working on establishing a new trial site in Sonoma County, Tales Vineyard. And these will be to investigate the impact and usefulness of applying mulch under the vine. Mulch will be banned one time, 2.5 to 3 feet wide strip under the vines on 1 acre block of Pinot Noir. The mulch will be layered roughly 6 inches. Probably half of the block will be treated with mulch and the other half with none. The entire block will be managed the same in all other regards. And just like at the other trial sites, we have collected soil samples recently in the tractor roads and under the vine in treated and untreated areas to establish baseline soil conditions. So this will help us track down any benefits to soil health, soil carbon sequestration. I also think that applying the wood mulch under the vine could help us understand its potential to mitigate weeds. So among all of these trial sites that we have been doing over the years, we're trying to investigate a lot of different sustainable management practices that have been popular or have gained some attention over the years among the North Coast vineyard industry. And we hope that this would allow us to track down much better what will work for a region, what wouldn't work for a region, and try to modify some of the things that we do to make it fit a little bit better into what we are doing in our region. And this is important, like I mentioned, because of soil variations, topography, and microclimate changes. So we are very interested to see how things progress over time. Like I said, a lot of these trial sites have been going on for a few years, and we have already made an effort to collect soil samples. So we will be able to share a lot more information with you in the incoming years. The next effort that the Soil Hub has been making over the past few years in relation to soil health research is soil health assessment. So we have been conducting soil health assessments in vineyards across Napa, Mendocino, and Sonoma counties. And these soil health assessments involve the collection of soil samples as well as some in-field testing. And I will discuss in a little bit more detail in a second what these soil health assessments involve. And the purpose of these soil health assessments is to determine how different soil management practices impact soil health. We are particularly interested in different management approaches for both the undervine and the middles in between vine rows. So for the undervine, we're looking at herbicides, use of herbicides, cultivation and mowing for controlling weeds and vegetation under the vine. And for in between the vine rows, we're looking at till, no-till, alternate till, and the use of various cover crops among these systems. So far, we have assessed soil health in 75 vineyards across the 3 counties and collected close to 300 soil samples. And we expect to collect an additional 130 samples this year. We have tried to represent different soil types, microclimates, topography, and different soil management practices across the 3 counties to accurately, or as accurately as possible, represent a region. And the process of conducting a soil health assessment also has an important educational component, as it fosters a farm discussion and interest in soil management, because the participating farmer is given a one-on-one technical assistance and an opportunity to discuss their own objectives, interests, and potential barriers to adopting soil health practices. So it's a great way to begin the conversation with growers about looking into improving and maintaining proper soil health. The farmers are introduced to the concept of soil health indicators and implications for meeting their own objectives. And this helps farmers move beyond traditional soil tests to a greater understanding of multiple soil functions and introduces them to emerging technologies of an integrated approach. We look at soil health in the same way, in a similar way that the USDA, Natural Resource Conservation Service, looks at it. They define it as soil health as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So a healthy soil is a soil that is able to do its function, and in the agricultural system is to be able to maintain proper crop development. The soil health concept recognizes that the soil is a living system. So this is what we're looking at. We're looking at a living system, and as such, we look at physical, chemical, and biological properties. And we look at these different parameters in order to determine how the soil is functioning. And the idea is that we will create some sort of scoring scheme, and I'll talk about it in a second, that would allow us to give growers a score of how their soil is performing, or how their soil health is in relation to the rest of the region, or to what we will consider a healthy soil. And we are doing this by sending samples that we collect to Oregon State University for a comprehensive soil health analysis, where they test the different physical, biological, and chemical soil properties. So this is how we're defining soil health. It's the ability of the soil to do its function in relation to all of these different physical, biological, and chemical parameters. So what we're looking into is texture, which influences water infiltration, the potential to store organic matter and nutrients, the aggregate stability, which influences water infiltration and oxygen moving to the soil, changes relatively rapidly with soil management changes because of the role of living organisms in the soil aggregation, such as fungal hyphae, earthworms, bacteria, etc. So it's important to keep an eye on it. Available water holding capacity, which is the amount of plant available water in a given soil profile. The biggest influence on available water holding capacity is the soil texture, with loam and clay loam soils having the greater available water holding capacity. It's also important to track down increasing soil organic matter, which as soil organic matter increases, available water holding capacity will also increase. pH influences nutrient availability, soil biology, and plant health. Electric conductivity, which is a measure of soil salinity, which it's not a big, it's not an issue as much for us here as it is in Southern California, but it's important to keep track of that. We also look at nutrients, NPK, calcium, potassium, magnesium, the whole shebang. Cation exchange capacity, which is important to determine the storage of nutrients, such as potassium, calcium, magnesium, positively charged nutrients. The total organic carbon present in organic matter, which is a sort of nutrients and improves soil structure and water holding capacity. And this is important because storing greater amounts of carbon in soil, i.e. carbon sequestration, it's an important mechanism for reducing atmospheric carbon dioxide levels and mitigating climate change. And organic matter is a source of food for microbes. Soil respiration, it's an indicator of soil microbial activity. Potential universal nitrogen is amount of organic nitrogen converted to plant available mineral forms under controlled conditions. Bulk density, which increases with soil compaction. Increasing bulk density is a negative indicator for healthy root growth and water and air movement in the soil. Water infiltration is measured to estimate the soil's ability to capture rainfall and prevent surface runoff and erosion during high intensity events. And compaction, which can restrict root growth and slow down water movement through the soil profile. So this is how we are defining soil health. It's a list of these different parameters that we are investigating. And the objective is, for this research, is to develop a regionally specific tool to aid in the interpretation of soil health assessment data. Currently, there's no regionally adjusted data-derived tools or thresholds or scoring systems available in California to guide management decisions related to soil health. So when a farmer sends a soil sample for analysis, they may get some recommendations from the lab, but those are more or less generic. They are not tailored, or they are not calibrated to our soils, our microclimates, our topography. So what we want to do is we want to utilize all of our data to develop a tool that will allow all of North Coast farmers to be able to get more precise soil data interpretations and the results that are tailored to their soil type, microclimate, and topography. So similar to what I have here in this scoring system right here, this is very similar to what Cornell University has been doing for the region. So anybody that is farming in the close to Cornell University in that region, they can look at this scoring system and see if there are different parameters that I described in the previous slide. How do they score according to that? So are they in the green, the yellow, the red? Do they need to take action? Do they just need to keep an eye on it? Do they need to not do anything? So we want to develop a similar tool to this that would allow our local farmers to be able to understand more precisely how their soil health data it's compared to the rest of the region, but also compared to particular standards. So this should allow North Coast farmers to have the necessary tools to help them improve or maintain adequate soil health. So we want to develop these tools to be able to help them make better, more informed decisions. And the way that we're planning to develop this scoring tool is by compiling all of our data we have so far from the 300 plus soil samples we have collected so far into a database that will allow us to identify regional specific patterns between the different management practices and soil health. We will then develop the scoring system similar to what we have here, and this will allow the farmers to interpret soil health analysis results and be able to make necessary adjustments. The tool
Miguel?
will be yes.
Yeah, we got a little under 15 minutes till question and answer time.
Okay, I'm wrapping it up in the next 30 seconds.
Great, thank you.
Yeah, let's see. So once we compile all of our data into database, then we'll be able to develop this scoring system. And our objective is to have the database and this tool developed by the end of next year. And in the meantime, we have begun analyzing the data we have so far to look for any patterns and to fine-tune the parameters we are analyzing. Our next speaker, Shannon Cappellazzi, will discuss in a little bit more detail because she's helping us out with this database development. So she'll be able to talk a little bit in more detail where we are with this and where we are heading and how our efforts compare to what the Soil Health Institute is conducting. So with that, I thank you for your attention. I think there will be time to ask me questions after our next speaker. So please check our website and let us know if there's anything we can assist with you. We're always happy to hear from you. Thank you.
Okay, I'm just gonna jump in and say that our next speaker is Shannon Cappellazzi. Sorry about the pronunciation there, but she is the lead scientist and research soil scientist at the Soil Health Institute and is familiar with our program because she started with us when she in her former job, and the title of her Well, here it is. Her title is Soil's Capacity to Function. It All Depends. Take it away, Shannon. Thank you.
All right, thanks, Keith. And thanks, Miguel, for giving that great overview. Yeah, one of the things that we have been thinking is that the old scoring systems that have been working for soil health, they really didn't take into account these different issues that we have in the West. And so, yeah, Keith touched on this, so I won't get too far into it, but one of the things that we really think about with soil health is that it really is about the continued capacity of the soil to function. And so as we're doing all of our work, we really key in on that ability to function, recognizing that it is a vital living ecosystem that sustains plants, animals, and humans. And so this is a major part of our approach, that thinking about that soil's ability to function. Another thing that we've really been thinking about is that, for a long time, soil health has been largely focused on producers, which is great. That's on the ground, what we need as far as actually having adoption. But there's a lot of other people throughout the supply chain who are also interested in soil health assessments. And so we're keeping in mind that, farm advisors and supply chain people and economists, they all also want to be able to use a soil health assessment. And the thing that I think unites all of these different people is that they all want a clear interpretation. And, that can be related to both economic and environmental outcomes. And so what we're trying to do is think about how we can make these assessments relative to the specific place and applicable, and comparable between locations, and then be related to those economic and environmental outcomes. So we've really been thinking about how each of these stakeholders will use the assessments. And so I think one of the things is, people want to know, is the management moving towards a healthier soil? And so one of the approaches I'll talk about here is that we're seeing soil health assessments for a long time are relative, right? You measure the indicators for a system that might be business as usual, what has been happening for a long time, maybe a more advanced soil health management system, and then also some kind of ecological reference or target. And so I'll talk a little bit about why we're using these minimally managed grassland areas as this ecological target for these regulatory and supporting services that the soil can provide. When we talk about the soil's ability to function, people ask, okay, well, is this soil actually performing each of the jobs to the best of its ability? And so we're doing this by trying to assess these individual functions that the soil perform. So we know that the soil both stores and cycles carbon. We need to put that carbon back into the soil so that the microbes have the ability to use that carbon and continue to cycle it, to use it, and to perform everything else. One of the other functions that is very relevant to production agriculture is cycling nutrients, taking that organic matter, turning it into plant available, or the nutrients that are on the soil minerals themselves, turning that into plant available nutrients. Another thing that I think is well depicted here in this fence line picture is this ability to store and deliver water. So here you see, the same soil, the same climate. There's clearly a difference in the amount of water that can infiltrate into that soil, and it looks like that main difference might be the residue cover, right? So having other functions that we will be working on, not necessarily in the projects we've done so far, but suppressing disease and pests, as well as being this reservoir of biodiversity. And so one of these things that we have been going towards is an approach to a simple suite of indicators. Miguel showed that entire list that gives a very comprehensive view of it, but we had this project called the North American Project to Evaluate Soil Health Measurements. And one of the goals was to be able to create a list of minimum soil health measurements that will be able to say something about the soil's ability to function without being so cost prohibitive that people can't use it as a regular soil test, that somebody along the supply chain can't use it for a purchasing decision. And so we are looking for how these different indicators can be used in conjunction to approximate specific functions, and how that can then allow us to better assess the direct and indirect impacts of management practices, the land degradation or regeneration. And by looking at these specific functions, we think that that's going to be able to allow us to better relate the benefits of improving soil health to the economic and environmental outcomes. So in this giant project, one of the things that we really wanted to do was understand how these inherent place-based drivers are influencing the soil's capacity. Like Miguel said, that there are a lot of soil health assessments have only assessed a small amount of inherent features, and we realize that there are many things that create a unique soil, right? So there are just these layers of interactions, the plants and the soil organisms and the climate and the parent material that make these soils unique. And I think we're all becoming more aware of just how pivotal the microbial community is in the soil functions. And so what you see here is a map with a climate moisture deficit, where the dark blue colors indicate that there's plenty of water, more water than is needed for crop productivity. And the warmer the color, the more severe that, that water deficit is. And each of these black dots are the 124 long-term agricultural research stations that we sampled in this large North American project, where we were really trying to evaluate these indicators. We had the privilege of actually using microbial, of doing full genomic sequencing, 16S and ITS sequencing. What you're seeing here is called a detrended correspondence analysis. Don't worry too much about exactly what it means. This is the bacterial and archaeal communities. And each one of these dots represents a single soil sample from a replicated long-term research trial. The dots are colored based on this Hargreaves climate moisture deficit. And in order to understand this, what you need to know is that the closer two dots are to each other, the more similar that overall community is, right? And so one of the things that we can take from this is that the overall community that is in that place is really driven by the climate, right? So we see these soils over here where we're in this hot, dry climate, low, high evapotranspiration rates, and the microbial community that is there, that is performing all of these functions, is very similar. Where we have these really wet soils, we again see these gradients, and obviously there's differences, right? There's going to be differences in the community based on management practices, but this is really an inherent driver of the microbial community. Another thing that you'll see throughout microbial literature is that the pH also drives the community. So here we have the exact same detrended correspondence analysis, where each one of these dots represents a soil sample, and the closer two dots are to each other, the more similar the community is. And you'll see a really clear gradient here going along the x-axis, where we have the low pH soils on this side and the high pH soils on this side. And this x-axis is describing 45% of the variability of the microbial community. So this is one of the tools that we are using to understand what inherent properties really need to be taken into account as we're trying to make this system. Another thing that I think is widely known by anybody who's ever worked with soils is that texture also has an influence on the soil's ability to perform these functions, right? So here we have a density distribution. We have about 2,000 samples overall in this database. And so looking at potentially mineralizable nitrogen and breaking it out into groups, the coarse, the medium, or fine, there is a statistically different potential to mineralize nitrogen between these textural groups. And so we think that this is an important characteristic that we take into account as we are determining how we are going to be able to interpret different soil health assessments in the context of the place where these soils really are. Again, with intact available water holding capacity, we see a significant difference between the coarse, medium, and fine textured soils in the amount of water that they can hold. And so, these are two of the functions that were most directly measured in our trial. And so we're using texture as part of that interpretation.
Shannon?
Yeah.
You got 5 more minutes, and then question and answer.
All right. I got a little bit of a late start here, so I'm going to skip this just a little bit here. This is one of the things that we did was we said, okay, what indicators do we really need in order to understand our ability to mineralize nitrogen? The final answer, cut to the chase, was that we needed soil organic carbon, nitrogen, and respiration, along with these site-specific parameters, the climate, the texture, and the pH, to understand how that mineralizable nitrogen changes in the system, instead of measuring necessarily mineralizable nitrogen, because that's an expensive, time-consuming measurement. So maybe we can just use a simpler suite. Using that same simpler suite, we've also reanalyzed these pedotransfer functions, where we use clay and sand and organic matter to estimate how much water the soil can store. This red line was the old pedotransfer function, where folks and they use the same parameters, but they were using repacked cores instead of using intact soil cores. And we see that when we use intact soil cores, we have this big difference by soil organic carbon, where something with more soil organic carbon holds more water. And so we have redone these pedotransfer functions, taking into account the soil organic carbon by matching that to what's going on in an intact core. And here, then, instead of having no difference by carbon, we have a big difference, which is much more reflective of what producers actually see. So we're using kind of this simple suite of indicators. Soil organic carbon talks to the direct impact of the soil's ability to store carbon. Carbon cycling, we are looking at using carbon mineralization or a CO2 respiration with a 24-hour incubation to measure that carbon cycling impact. Aggregate stability is well related to erosion resistance. And so we have kind of this minimum suite that we're coming to, where we take into account that texture and pH and climate, and then have this minimum suite of measurements that helps us with these direct functions, as well as being able to talk about water storage, where we use those to assess water storage. We use those to assess nitrogen cycling. We also measured water movement, the saturated hydraulic conductivity as it moves through the profile. We don't have a good relationship to that one yet, but we're working on it. So to speak to this idea of the soil health target, I'm not going to get too much into the details here. This is a site in Oregon. On the left, you see what the soil looks like, business as usual after a hard rain. On the right, you see this reference perennial, where you've got this perennial grassland, these beautiful aggregates, tons of roots. Same day, right after a hard rain, and there's no ponding. It's easy to walk on the surface, and there's plenty of water movement. So we have that business as usual in the wheat fallow with more moldboard tillage reference site. And then we also measured a soil health management system. And here we had this wheat pea rotation instead of just wheat fallow, and we had no tillage. And this flow, this is that saturated hydraulic conductivity. So in this business as usual, we only had 1.5 centimeters of water able to enter per hour. In the reference, we had 32. And in the soil health management system, we're getting there, right? We're making progress towards that ecological maximum for that water moving into the system. It was at 12 centimeters per hour. So we can extend this also to some of the other functions that we want to look at. So we've got this capacity to store carbon, and we've got this sliding scale where the business as usual is at 0.8, soil health management system was at 1.7, and that reference was at 2.2. Similarly to the capacity to mineralize nitrogen and the capacity to resist erosion measured with aggregate stability. And so one of the things we're thinking about here is that soil health management system, if we have those end caps, we have a business as usual and we have a reference, we know how far along that chain we've made it. So they've made it 65% toward that ecological maximum on that soil's natural capacity to store carbon. With mineralization of nitrogen, this soil health management system has made it 43% of the way there. With resisting erosion, it's made it 77% of the way there. So this is part of how we're thinking about it. We take into account these inherent features and understand how these indicators can tell us about how far along we've made it towards that ecological maximum. And so one of the things that we have done and we are doing, and this is a nationwide stratification plan, but it is relative to what we're looking at in California here. So here we have the counties of interest. Mendocino, Sonoma, Napa, Marin Lake, and they are colored by this soil strata. So instead of having every single map unit on here and saying that every single one would be different, we would never get to a big enough data set to be able to say something meaningful about every specific soil map unit. But what we have is this soil strata that are categorized in 3 ways. One is kind of that activity. In the legend here, you see plus, minus, BS. That is kind of this activity level, and then texture, and then the drainage class. And so each of these colors on here is something that we want to be able to sample. We want to be able to sample each of these strata in the different areas. And so if we zoom in on just kind of Napa, a little bit north of Napa, we have now, instead of having 200 different map units or soil series, we have about 10 different ones that we want to measure. And if we can measure the reference state and the soil measurements in each of those areas, then we can't, for that specific soil strata, we can compare any production system to that reference and get an idea of how far along the chain we are, so that we can have this relative scoring system that makes sense for the place, the soil type, the microclimates that you are in. And so, like I said, we've been working on this as far as a reference idea. We're rolling it out in Washington and in Texas and in Arkansas and in California, Idaho, Wisconsin, and New York. So we are ground truthing this right now. And we are hoping that the next year, when we are presenting this data, we'll be able to give you information based on the specific strata that the vineyards are in, so that we have this relative type scoring system. And just to kind of give you a heads up, we are looking at having this minimum suite of soil health measurements that really, because of the redundancy of some of these other measures, this really tells us a lot about what's going on in the soil's ability to function. So using soil organic carbon, respiration, and aggregate stability, comparing those to a business as usual and an ecological reference, where those soils are in that similar climate and inherent soil properties, is going to be able to give us quite a bit of information here. And I'm sorry that I had to move through all of that so quickly. I am happy to answer questions. I thought I was going to have a little more time, but I'm happy to answer any questions here or later.
All right, I'm going to jump in, Shannon, and say you did a great job. Sorry you got a little bit rushed there, but you actually presented the material really well, and that makes a lot of sense. It's really exciting to see what's happening, and I think it helps us who are involved with the North Coast Soil Hub and beyond to see the context of what we're doing fits in this much bigger picture, and that just makes it that much more exciting. So I thank you for that. I do want to say a quick, before we finish the North Coast Soil Hub talk and with the question and answers we have with the time remaining, a shout out to Charles Chembray, who I noticed is on. He helped get this going. He used to work for Napa RCD before Miguel was there, and he helped put the grant together. And a lot of what you just discussed was something he really helped lead the charge on, did a great job. Unfortunately, we lost him, and he went to work for the University of Nevada, Reno. But Miguel has come in and done a great job and with a different perspective. So thank you. And with that, Katie, take it away.
Sure, yeah, I'll try and get as many in as I can. I think this would be good for Miguel. We have a question about the timing of the annual soil health samples. Could you speak to that?
Yes, so we have been making an effort to collect our soil samples during the late winter, early spring. And our motivation has been mostly soil moisture. We want to make sure that we are consistently collecting the soil samples with sufficient moisture, but that also makes the sampling a lot easier. We will be going in the middle of the summer when the soils are completely dry. It'll be very difficult to actually extract the samples. So late winter, early spring. So around this time is when we are actually out there collecting the samples.
Great, thanks. And Shannon, could you speak a bit more to what you would like to see for these reference state samples for soils? Is this like a naturalized vegetation system, or what are you looking at?
Yeah, so there is the potential to have a little bit of variation. One of the things that we're looking at kind of as the first push is a minimally managed perennial grassland area. What we have seen is that, the grassland really epitomizes a lot of what we are looking for in soil health, right? It's covered all year long. There's often quite a bit of diversity. There's a lot of roots going deep down into the soil. You know, it has all of these soil health principles built into it. We recognize that not every site used to be a grassland, and so maybe it's not its native ecological principle, but we can normally find something where there is a grassland, and in our soil health assessments, we often see these highest values from these grassland systems. And so we're using that as somewhat of a reference or a target for what we might be able to get into, recognizing that there's also the potential for some of these numbers to go even higher. I have measured a lot of systems, especially when you get into a grazing type system, where you have considerably higher carbon storage potential, higher water infiltration, higher water storage, because of that added organic matter. You know, if you have a properly managed graze system, I've also seen the worst soil health scores in a poorly grazed system, but in well-managed grazed systems, that's where we see our best soil health assessments and our highest amount of ecological potential. So there's kind of this native ecological idea of the capacity to function. And then we know that you can move beyond that as well. And it's, you know, then we want to kind of also not go to the point where we are losing more nutrients. You know, sometimes if you get a really high nitrogen mineralization potential, you're going to have more nitrous oxide emissions and more nitrate leaching through the groundwater. So we want to understand that this is not always a more is better. There is kind of this ecological optimum where we see these tight cycles and things really working together to benefit, you know, that plant health and soil health of the system. So one of the other things that I think would be great in these targets is if we can get these targets for multiple native ecological sites, and maybe that will be a project that we're moving on in the future. To start with, we're starting with these minimally managed grassland perennials.
Great, thanks. And could you follow up too with what is the depth of soil sampling and why do you use that depth?
Yeah, so for a traditional soil health assessment, we use a 6-inch or 15 centimeter sample. In all of these reference projects, particularly because there is this emphasis also on soil carbon storage, we're going to 30 centimeters or 12 inches. The reason that it's been top 6 inches for a long time is because that's where the majority of the biological activity is in a lot of row crop systems. If you get a good grass system, you can get roots down deeper, you can get plenty of biological activity down deeper, but there is always a gradient. And so we start out looking at that top 6 inches. Hopefully we'll move deeper down into the soil as we move forward.
Great, thanks. And Miguel, how about you for this one? Are our California Ag Labs set up to process soil health samples and interpret them? And if not, what are we doing about that as a soil hub?
That's a good question. So we partnered up with Oregon State University because they are a research institution, so their lab follows research protocols. I wasn't here when the conversation started and why we went with them. I've worked with other labs. I mean, I've sent samples to other labs in California. And the problem is that not every lab is equipped to do the suite of assessments that we want to do as we're following changes in soil health. And the second thing is that currently there's no regulations really checking in on these commercial labs to see how accurate the results are. So because this was going to be a very research-driven project, we wanted to make sure that we have results that are accurate and that are going to be reliable. So that was our decision making process. There's other institutions. We have UC Davis nearby that also has a soil health lab that will be able to do a lot of the same suite of sampling that we want to do. But we found that Oregon was one of the few that will do everything in just one single sample. So we just send them one sample. If we were going with local labs, we would have to send different samples to different labs to be able to cover everything that we're doing. That's not necessarily what growers will need to do. We're doing this because we're covering more parameters. But I mean, local labs here will give you a lot of information as a grower, this was more of a research project, so we wanted to cover more.
One thing too I would say about that is that we are working on a proficiency program for soil health tests. A lot of soil labs participate in proficiency programs so that if you send samples to one lab or another, they're at least relatively similar. It has been a problem with soil health tests because there has not been a standard operating procedure, and there is quite a bit of variability in how samples are handled. And so that is also an active area of work that I am participating in, is that lab standardization of soil health. And as more and more people are interested in soil health, there are more labs that are doing this now. So, when Keith contacted me 5 years ago, there weren't that many labs doing soil health at all. And now I'm working on, some new large projects, and I was able to get bids from 5 different labs that would perform the full suite of measurements that we were looking for. And so, it's exciting to see more labs taking this on, and I will say, none of them were in California. So hopefully we'll get a good California lab on board once we have the standardization and people have a better idea of the reliability of these tests.
Okay, I'm gonna jump in and say we have time for 1 or 2 more questions, and then we're gonna go just about 2 minutes late. The Zoom won't cut off at noon, so we're just gonna squeak a little longer. And Katie, whatever you think is best to ask.
Sure. Yeah, I think people are interested in learning more about how you're actually collecting the data. Do you have a team, or are you collaborating with other entities? I think maybe both of you could speak to that a bit.
I would say both of us probably have teams, right? So as far as ground truthing, we have hired quite a few different project scientists, and we're going out and collecting samples. We're not just relying on others' data sets. We want to make sure that the samples are collected in a uniform time, uniform time of the year, which we try to do as close to, before spring activities start as possible. And so we have a whole team of different people going out and taking samples using intact cores for bulk density and intact available water holding capacity, as well as augers and spades and soil knives for collecting the surface soils.
You have anything to add there, Miguel?
We do everything ourselves. So each RCD has a team that goes out there and collects the soil samples. And we partnered out with Shannon to help us with the data interpretation. So that has been an approach. We do it in-house, and then we're going to be working with Shannon to figure out what the data means.
Excellent.
Great.
Well, it is noon, so should probably just share a couple closing thoughts, and then let people get on with their day. But Shannon, thank you so much. Miguel, thank you. That was great. And Shannon, it's really great to see that you moved from OSU, where you were a great partner, but to soil health, and you're playing a leadership role there. And it's I've been seeing the work you're doing and that your institute is doing. It's really great. So thank you, and thanks for sticking with us and keeping it going. And it's good to know that things are actually, there's more work being done. It's not just a little here, a little there. It seems much broader. So this has been a great day. This has been really exciting. I personally cannot wait to go back and see the video. There's so much information I couldn't keep track, but as well, as a coordinator, there was some behind the scenes action, and there's just a lot there. So it's really exciting to have all these folks present and really get some really the brightest minds on this involved. Thank you to all the participants for coming, and to all the presenters, and for the NRCS and your support. A lot of good information there. Stay connected, check in with your RCDs and this North Coast Soil Hub to know what's going on. We do have another full day tomorrow, and it's going to be more grower on the ground focused. So we invite all of you to come back for Shop Talk, Soil Health in the Field. It's the same link you used today will work tomorrow. And tomorrow we will feature an Under the Vine grower panel with 3 different growers from the area. There'll be grazing in the vineyard from Kelly Mulville, who has a lot of experience with that. We're going to hear from Dr. Cristina Lascano talking about soil biome and how important the soil biology is to that. And she has a lot of the new information and what's relevant to good production in vineyards. And then we'll have an economics and marketing panel with 4 different growers and 4 different businesses that use their environmental and resource stewardship. They integrate it into their their marketing and are using the types of practices we're talking about today, not just to only to benefit their production, but also then to benefit their bottom line. So thank you very much. Stay in touch with your local RCD or equivalent, and we will see you tomorrow at 8:30 a.m. Thank you very much.